Okay, good morning to everybody. Welcome to Schmidt Ocean Institute Dive 746. Thank you to our YouTube participants for being patient because we were troubleshooting going live on YouTube. So you're tuning in a little bit late. But we have returned to Mocha Island Seep site, the deeper site. We are at 2,742 meters, and we decided to return to this site because we found this beautiful bacterial mat at the end of the last dive, and we wanted to revisit it. And so we are now in the midst of doing push cores. And yeah, so all you missed was us just relocating to find this bacterial mat and putting the first few push cores in. My name is Juliana, and in Mission Control, I have Doug and Tyler piloting. I also have Aaron as watch lead, and in the back row is Adam, Maurizia, and Dan. Aaron, do you want to talk about why we wanted to come back to this area?
Yeah, of course. So as microbiologists on board, we're super excited to find some bacterial mats. Bacterial mats are a really great flag of some really exciting geochemical microenvironments at the sediment and below the sediment. These mats generally indicate some sort of sulfur oxidizing mat. And suggest that the microbes here are taking really great advantage of the methane in this region. So for us folks interested in seeing which microbes are consuming the methane, the rate at which they're consuming the methane, how they're arranged, what the methane concentration is, all that jazz, these are always like really promising sites and very exciting finds for us.
So do you process these cores in a different way than, say, geologists would?
Yeah, so for these smaller push cores, or small relative to the huge gravity cores that we send down as well, we slice them up into these little hockey puck, bubbles, and these thin slices, kind of like hockey pucks, and then, one sec, before we start pulling, can we also fire an instant at this site as well?
Do that right now while you're doing your spiel.
Cool, thank
No
you.
problem.
Yeah, so once we have these hockey pucks, depending on what we're after, be it the geochemical environment, the microbes, and their taxa, some of it we use for incubation. From there, after this horizon, they'll all be sent to their different fates. What seems like millimeters to us is relative to a microbe miles and miles. So even just in the seemingly small distance from the top of the core to the bottom of the core, we can see huge variations in the sort of activity going on in these microbial communities.
This kind of one is fire.
Thank you.
Just want to make sure you have your map drawn.
Yes, I do.
All right. we're gonna start pulling.
I'll get one more frame grab in case, but it should be good.
Good?
Yep.
All right.
Thanks.
Sweet. Come out, porch. Two views, nice and easy, that's not going anywhere.
Yeah. And this one's a little bit misleading because the surface seems like a little bleeding like stuff. But it looks good.
I'm just gonna leave the porch here.
Looks
You got a good view to pick them up,
Yeah.
both places.
Smooth.
Oh.
Oh, not gonna stay.
Dump it.
Yes. If you could shake it out and maybe reinsert it near 7, possibly. Maybe behind 7 to avoid some of the sediment that's falling out of it.
Got it.
Nah, you're fine, that's perfect. You got plenty of reach. Right where you're going, perfect. Yep, you're in.
You guys wait while I grab the other one?
Yeah, you might as well.
Found some really deep black streaks. Or if it's been pulled down, I guess when the push core went in, actually.
I'm sorry, Aaron, are you asking a question or were you just making?
I was just
Okay.
thinking out loud.
So for each of these sites that we've been visiting on this cruise, we're not only just interested in ROV dives, there's also a bunch of operations that happen when the ROV is on deck. So for example, every station we have several CTD casts to do, as well as some gravity coring and some net tows. So we'll often take that opportunity when Sebastian is on deck for other operations to be at play. This gives some scientists time to sample. It also makes sure that we're not just having downtime with ship time. And then we'll always just take the opportunity to continue mapping in the area, just because we have the resources, and it's a good use of ship time if our scientists are busy. On one of our first few cruises on Falcor 2, I think we were mostly mapping and doing INSAS with the ROV, and we stayed down for, I think it was almost 3 days straight, which was almost a record, I believe. But a lot of our cruises were just ready to come back up, and a lot of these dives were just ready to come back up, and the porch is filled. A lot of ROVs also utilize landers or elevators, where it's a physical piece of equipment that you deploy from the ship. It lands on the bottom. The ROV
Yeah, I
can
think we're
pick
good
it
on
up and
push
move
course
it
here.
wherever it wants.
If we want to
And then
look
we
around
will
a little bit,
then fill it with samples. And so when the ROV is ready, they can release it to the surface, we can pick it up, and then that's a way of keeping the ROV down while still getting the samples back to the surface. But we haven't really utilized that. We do have a lander on board, but it hasn't really been a request from some of our science parties. There's been, in terms of autonomous mapping, Sail Drone has done a lot of work to deploy these sail drones in the ocean that have solar and wind power to propel themselves in different areas of the world. They often have a towed sonar array to map as much as they possibly can. So I would love to see way more out there, but there's definitely some autonomous mapping examples.
Shout out if you see something.
What more of that Xenophyophore that Greg described? I don't think he needs any more, but it's a lot more than I expected. mound that we saw yesterday, or?
Yep, this is coming up on the mound we saw yesterday, I believe. Get the map correctly. We do see bubbles. Did Jeff wants a gas tape?
Yes, I think so. Oh, they're right there.
Bubbles.
Yeah. I just saw some right here. Oh, there's another kind of to the right. Not like a very consistent stream yet.
I think there are probably places it's going to be coming out more effusively.
Just keep looking around.
Yeah. I think if we can find even the same stream as yesterday, I think he said he didn't mind.
It was pretty obvious yesterday.
Yeah. What's
That was
this?
over to the left.
What is that?
Left from where we're at.
I think it's a new cucumber that we haven't seen yet.
Look at that.
Yeah, I think Greg would probably be interested. If you could slurp one of those. Or if the only one, it seems.
Which jar would you like that?
Don't know that I have a preference, and maybe I should, but
Yeah, I'll just give you a little background. Some of them have screens in them for catching small animals. So probably for this, you're
Of
gonna want
course.
one, either no screen or a larger screen.
Right. Cool. We can just do 5, 6, or 7 then.
Okay, cool, problem.
Cool.
We're on 7, so.
Alright, 7 it is. Alright, I'm set down, Tyler. I'm gonna kick that rail out.
If we can just zoom out for a sec or pan
Sure.
down so I can
Yeah,
grab a
sorry.
frame grab first. No worries.
Wow.
Such a cutie. He actually truly does look like a pickle.
Yeah.
The most
Yeah.
cucumber-like cucumber we've seen.
Typically you get some tape there.
What are the little waving?
Could we also put the lasers on? That seems super.
Yeah. Good call.
That's it all the way out. Let me just get a measurement real quick before you grab that, Tyler.
Just work off the.
Could like,
Big.
yeah, like 18 maybe? 15, 18? Whew.
Is that a good shot for you with the lasers?
Yeah, that's perfect. Thank you.
Yep.
Got it.
Alrighty. Push it, please. Still pushing it. There you go.
Drag this over. Next one.
Let's pick a pickle. Pick a pickle.
What is the tongue twister that goes to?
Say again?
Isn't there a pickle tongue twister?
I don't know. Yeah, I think it's like a Peter Piper.
Yes, that one.
Peter Piper, pickle or something.
Eddie?
Is possible to collect the,
Ready?
the snails
Red,
Yeah.
in the same jar, maybe?
Okay, cool.
That section.
If while the suction's out, we could also grab these two snails. If they'll fit in the jar with that absolute unit.
Oh, barely fit in the jar. Look at that thing, man. Holy cow. I'm not gonna lie for a second. I'm like, oh, that's the tube stuff. All right, you see the snails there? That one right out there, and then the one to the left.
Just keep it in there. Can't really see my buggy, but
You're looking all right.
yeah, the one just up here, right?
Yeah, the other one I might have to move for. I can move for both actually.
Yeah, let's see that.
So I
I'm
can
pretty
pull that
stretched
in.
out.
Yeah, just pull that in and I'll just move. It's not a big deal. All right. You happy? It's so nice that I'm still light.
Yeah.
I can actually move without stirring everything up. Mouse to grab. Okay. Selection's on.
Oh.
Other side.
How did that cucumber fit? But this is
Back on. That other snail never came through either. Or did it? No, I don't see it. Something back on. Probably got to get that a little bit shaken.
Yeah.
Erin has just been able to access YouTube.
Yes.
Okay, good.
Yeah.
Hey, Joe. Shout out to Jeff.
Just look at the end and see if it's stuck. Yeah, I don't see it stuck in there. Definitely didn't come through the tube.
Lots of mounds.
What's that?
I was saying lots of mounds. Mounds. Hi. I have an animal for you to ID for sure. No clue what it was, but yeah, very cool. The bright green cucumber, I think it was a sea cucumber, and it looked like a literal cucumber.
Yeah. It's like some cucumber.
Yeah, we got one of those. Shockingly fit through the yeah, so we got all the macro scores. I was doing one final scope in the area for any anemones, corals,
So
sponges.
you got 6 for you guys.
Yeah.
Plus,
We got
Some-
5 for us here. We're gonna hold on to one in case.
Okay.
One, Davide only needed one. So
he told
I-
me none yesterday, but after I submitted this.
Okay.
But,
Oh, yeah.
but
We try and do a gas station.
you're good. You were gonna pick one quote.
If there is one, I think one.
Yeah.
yeah. And then I got
Do you need more? Because Eulogio's not taking any today.
No, just one.
and the one for Pachi, all in so
Okay.
8 total in the mats
Do you
and
have-
Niskin.
Do you know?
And then we did get one of the Xenophyophores.
Xenophyophores. Yeah.
I don't think any orange stuff got in, but I have a small sample that it can at least you could at least sequence see what it is.
Is it? It's not a big one that fills the tube.
We got it. We were afraid of breaking it if we suctioned it, so it was in one of the second push score for Davide that he didn't need. I repurposed for that because I was afraid of breaking it if we
Okay,
suctioned
but is
it. Or-
it big?
It takes up a pretty much
whole
a bit
thing.
before, yeah.
Yeah, they're really fragile.
Yeah. Yeah.
Okay, great. Do you want to assign? I know
Oh.
he loves to have these.
Yeah, I wrote all these on the side. Ours just because they work together. And then all of these
No.
honestly look really great. A lot of bubbles too when you're pulling them out. So I don't know if anyone's preference, but they all looked pretty similar and pretty deep. So I don't think you can really go wrong. But 9, 10, 11 will be for the 3 of them.
Touching.
Pachi, Davide, and Jorge and Baird.
And are we back on the big bubble size?
Yes, I think we're probably sampling for Jeff Beeson with the gas sampler here. And then after that,
Basically what you have to do is
So-
wind up that one, just drop it.
So you've been going south since the bacterial. Right.
Grab
Yeah.
the orange
But
monkey
not-
fist,
Yeah.
the starter,
Yeah.
bring it out.
we want
Face
to
it.
find bubbles first for Jeff.
Grab the gas tank, port, lock it, get it ready, like over starboard, bring it over the bubbles, flip it, funnel. Pretty easy.
Thanks so much.
Want to take the port out a little bit?
I will do that, thank you.
Are there acceleration?
But it's a tight fit up front, so. Like, I'm gonna stop there.
So the only place we.
I'm gonna bring the camera all the way back, so you can see everything that's going on. So, just pick that one up. You're gonna have to come from the top, or you'll be in the middle of all the push cords, kind of just like you're doing right there. Yep, just don't come too far, you'll hit the NFL possibly. No, you won't, you got plenty of room there.
Here, there's a zone.
Oh.
Where the seafloor, it's much rougher. And it looks like some sort of mass transport sort of thing. So
Yeah.
if there's time,
Yeah.
we'd like to go over there just to there's not part of the seafloor, but that could be.
Just pick that up, bring it out.
You want to
That way.
map it?
Yep,
or-
keep going. You don't wanna hit that plunger.
See what it looks like. Okay,
The
we
gas
up, you
tank's
know, the
pretty much
dive
useless.
is coming up at noon, so it's not a.
Anywhere
very
there you can drop it,
long-
has no
I'm
tension
just
on it.
thinking, what, Davide, is there anything you need to see?
It's a real bear to pick it up,
Else?
because that monkey's fist isn't doing anything it's supposed to.
That
So
spot.
now, if you wanna grab the orange
And then
monkey
what about
fist.
the, all the way north bit?
Yep.
We never explored all the way to the top of that. Is that something?
So just watch grid 8.
I think I said that point, I saw.
Split the
We
back.
got about halfway through that feature.
Yeah, just grab
Yeah,
it.
that yesterday.
Yeah, don't go too far down and you'll be on the pipe,
Wasn't
which you are.
watching, so I don't know exactly what it.
On the pipe?
Yeah, you see where you're gonna hit the, what you call it?
Oh, I think I'm a
Yeah.
little above that,
I think
right?
those would be two
You're
priorities.
just touching it with the yellow.
Okay.
You can grab it there.
I mean,
There you
I
go.
know we've achieved already the
There you
number
go,
one
yeah,
priority.
and then
with
just come out
Dive,
nice
which
and
is to
easy.
get microbial methods.
No,
We're still working on that.
You just
It's
don't
the
wanna
only place
hit the
we
plunger.
can do it. Yeah.
Yep.
We didn't have any
And you don't wanna
pictures yesterday, but it.
move the funnel.
Yeah.
There you go. And then just start swinging it out.
But,
Out.
yeah. Thank you guys for taking the lead.
And just spin it so that the handle faces that way. Yep, just like you're doing. And then just get it in a good spot, out a little ways.
Do you,
And then, yep, right there. And then you can go ahead and
guide them?
freeze there.
They-
Well, they know what you're.
Drop the port arm. What I'm going to do is I'm going to retract it, make sure the gas tank is fully retracted.
Cool.
It's a shame the funnel isn't facing the other way, you can just do it right here. So, and you know where you wanna grab it is in the T slot, where the T is.
Right, that's
Do where the T is.
Get the groove.
Yeah,
And then remind
both
me,
grooves.
Locking the jaw, what do I hit on here?
Yeah, you can lock the jaw.
Which buttons do I hit for that?
Good morning, everybody.
Operating, yep, jaw, it's on
We're
position.
having a scientist shift change here.
The next position is locked, but don't. Can you verify that's locked? Here's both your pictures. You sit, and there, and your side. There you go, right about there. Start closing. Just close it easy as you're going. You don't want to go too far. You want that in a slot.
Yeah, I gotta get it across.
Yeah, there you go, right there, perfect. And start closing. It will start to self-align, right, like that, and just close it all the way. Just hold it closed. Close it all the way, perfect. Now hit that button. Locked. Okay, now lock your arm. Yep. Okay, and now what you want to do is with the starboard arm, just let the pressure off of the monkey's fist come down, and then once you see that the other arm definitely has a good hold of it, then you let go. Yeah, just come down easy, you'll see the slack come in the rope. That's perfect, you can let go now. Yep, and then just move that arm over to the right, out of the way. Perfect. I'm gonna porch in while you're doing that. And once you're good there, then just fire up the port arm.
We have, this is Lisa Levin from Scripps Institution of Oceanography. And we have Alice Santiago, data logging, and Pachimunos as co-lead. And we're all listening to a lesson on how to do a gas tight sample to trap the methane bubbles and bring them back for analysis of, I guess, stable isotope analyses, and what are the other things that are being measured? What else besides stable isotopes?
You don't have to get
being
on the
analyzed?
seafloor.
Oh,
Just get over it so that you catch bubbles.
helium
Yeah, you're already
isotopes.
catching bubbles. You see what I mean? There's so
And
many
the composition of
bubbles,
the
they're
gas.
almost half full already. This is such a stream. So you just want to hold it like right there. Yep, and I'm going to zoom in so that we can see the fills. And then I'm also going to zoom in to sit, so that we get a good picture and cut through the cylinder when it starts to get ready for that. This thing can be filled
This
up.
is a really
It's already way full enough.
nice, active bubble site, filling this funnel very, very quickly. Or filling the sampler, I should say. And what you're seeing are gas bubbles sticking to the side of the funnel forming gas hydrates or frozen methane. But much of the gas is entering the sampler itself.
So that's enough the other day?
Is that enough?
Yeah.
Okay. All right, I'm gonna go ahead and actuate.
Alright, I'm still frozen.
Is someone walking this?
Yes, red and green tape.
Huh?
Red and green tape.
Yes, thank you. Gonna catch it?
I don't know, it doesn't look like it is. Oh, why is that not aligned properly? Retract. You might have to rotate it touch. Why is that not aligned?
I don't know, but I can just go like this.
Yeah, you're gonna have to pitch up and turn so that it aligns with that. Yep. No, turn the other way. Yeah, as you pitch up and keep going. Keep going, keep going, keep going. All right, it should be aligned now.
I'm frozen.
Okay. This one out. There we go. Now it's pushing through.
The gas hydrate that forms here, it's also sitting on the other side of the rock, it's highly flammable, for sure.
Can you move
Somebody
that
in the
over?
chat was asking about tube worms and succession. We are really uncertain why we have seen no tube worms yesterday or today when we've been at more than 2,700 meters water
Can't
depth.
really see it, you're right.
At a 2,200 meter site. We had lots of tube worms 2 days ago, so we're unsure why they're not present here, and what the specific requirement is that's different. I had the thought that perhaps we are in a deeper water mass,
That is all the pressure I can apply to it.
Antarctic water, or some water mass that doesn't carry tube worms from the
Definitely see it's moving.
polar region,
But I can't see that
Antarctica,
already.
but it's unclear.
Bring it across a little bit.
Tube worms do not apparently require as much seepage and methane as the clams. These really big clams, which are Archibesica, potentially Archibesica gigas, but we're not sure of the species,
Yeah.
Good to go? Yeah. All right.
Oh, yeah.
Are
That was just in an odd
clearly
spot. You can see it.
loving the
All right,
fact
cool.
that there's
All right,
methane
so
everywhere
next
here.
steps is, you can flip that over, let the rest of the gas out, it's not a big deal. Yep, and then grab the monkey's fist, opposite of what you just did. So grab the monkey's fist, make sure you got a good hold of it. There goes the ice.
Oh, that's so cool.
All right.
It's frozen.
That was gas hydrate escaping from the funnel on purpose because the gas tight sampler is closed now.
We're going live.
Yep, just get a good hold of it.
It's frozen.
Okay.
It's locked. Unlock my jaw.
Yeah, fire the alarm first. And it's gonna go through another, cycle. So, go ahead. Nope, it won't go until you
Okay,
unfreeze.
that's awesome.
So you unfreeze. Okay, now hit it. That's toggle one more time. Now you got position. Just like you did before. So you can just come down, get the tension on the, a little bit of tension on the rope. There you go. Now you can let go. There you go. That's it. and then just move that arm out of the way, you can stow it.
Let go more like wing growth. Wing growth. Wing growth.
Good morning, John.
Yep.
Support arms still good.
Back on the camera, I'll let you lift that up a little bit.
Got it.
Same way, put it funnel in. I want to rest it in that little pan there, so I'm gonna go ahead and kick the porch out. There you go. And just set it right in the pan. As far as, yeah, you got bubbles on the camera.
Nice.
Dang you, Tyler. Yeah, just start coming down right there. Good. That's it. Alright, now's the fun part.
Oh, yeah.
Yeah.
Chasing
You see the,
monkey's fist.
you see the monkey's fist? Yeah, exactly.
Yeah.
Exactly.
It's getting out this wonderful
So,
T4 camera.
now you got it. You can kind of see it.
Oh, yeah.
See it?
Yep.
But you have to be careful of those push doors while you're coming through there. Just what you're doing. Just come in. I can try maybe, but nice easy movements before you hit next.
Yeah.
That's not helping you at all. I can try retracting the porch if you think that'll help. Because you're just about on top of those, this push door here.
Yeah. We got a little bit of a view. T4 camera.
Joe, when he's moved over here.
That was alright. Thank you very much.
All right, don't go down anymore. You're on top of 12. So if you can't lift that in there, some pitch, and grab it, you can make a blind grab too and see if you got it right there. Right there, you're touching it, grab it. Nope. You got it. All right, yeah,
Just
looks like you got it. Yeah, just come on up. And you got a good grab on it. And then just set it over the top.
Whoever invented the bungee cord probably had no idea of their value for deep sea research.
Oh, you dropped it.
We use bungees to strap in a lot of things
I can
sometimes.
bring the porch in a little bit, that might give you some relief there. Okay.
In all our
You
gear
can see it a
on the
little bit
ship
better, too. But you just gotta watch
for
that.
rough rides to make sure that the boxes don't roll around the floor.
You have to kind of use your elbow to come down, not your shoulder.
Gotcha.
There you go.
That's the way.
Yeah. There you go.
That's the way.
And on top of the push cord.
Next.
Hold those bubbles so we don't have.
They're coming out of the sampler, out of the gas
Well,
tank.
now you're clear of that push cord, come down on the shoulder.
Yeah,
This is gonna give you a better advantage, Tyler.
Yeah.
Yeah, you got plenty of room now. Just don't come to the left. Just don't come to the left. Come down, but you can't come left.
Yeah.
Much. Right about there.
I was on top of 5 for a second.
Yeah.
So it's really threading a gap here.
Just a little bit more.
Yeah, I think I'm high.
Yep.
Okay.
Come in just a touch.
A little bit on 12.
There you go. Yeah.
5. just on the
To your
road.
left, just, yeah. Try closing there. There you go. All right, now try bringing it out nice and easy. That's a much better grip. Okay, yeah, you're looking good there, buddy. Just bring it over to the
Did you
top
read?
of that. Over the top of the handle as well. Perfect. That's it. Rip.
Do you think we should take a nest in here?
All right, perfect. Nice one.
Thanks for the eyeballs there, Doug.
Yep. All right. Put it on the porch.
They took one already.
No. Would you like a Niskin here?
So,
Okay.
just to be on the safe
Yes, can you
side. Yeah, so we'll take it this one too.
Just went blank.
Yep, there it
This one
goes.
too.
Thank you. I would like to take a look at the fish that's been patiently sitting just beyond our work area. Pretty sure that fish was there yesterday. I don't know what it is.
Which fish is this? This one over here to the right?
Yeah, could we just zoom in and take a
can.
look at what's going on? It looks like, to me, like some kind of cusk eel or some, but I'm not really sure. And whether it could be a new fish. We often find specialized fish in chemosynthetic ecosystems. It looks a little bit like the kinds of fish that hang out in tube worms, and it's at vents and seeps, but in other places, but different. It's alive. It's moved since we've been here, but not much. Looks like there's a lot of scars on its body. Could you zoom in on the head and eye and mouth, please? Just, yeah, if you can grab some frames.
That's as tight as I can get without moving.
Okay, that's good.
Happy with that.
Have we seen any others of these? I think, do you remember seeing some yesterday anywhere else?
I think I saw them yesterday, just only one.
Just one.
Because it looked like they on video for 30 days.
It doesn't look particularly common out here, but maybe these images will be good enough for somebody to help us identify this or decide if it's new.
He's gasping for oxygen at that 42%.
I don't know, 42%'s not bad.
Yeah.
That's the oxygen saturation down here. 144 micromolars. molar.
Big mouth, big yawn. Good morning.
Maybe geology.
You have to wonder about the eyes and what the fish is seeing, because it's really normally pitch black down here. And maybe there are some bioluminescent organisms.
I wish he had eyelids right about now, with us down there.
Yep, he does. Can we just take a quick look at the tail end?
Sure.
Just to see if there's any features of interest. Definitely eel-like. The other creatures you see in this view are caprellid amphipods. We find them all the way from shallow waters. They're common in kelp forests, and I don't know about the intertidal, but they definitely live subtidally all the way from shallow water to very deep water. We're not sure what they're eating here. It does look like they're filtering. They don't seem to bend all the way down to feed on the bottom. They seem to be attached to clams, but also to the sediment. Maybe they are bending down and feeding on the bottom, picking off bacteria. You see sabellid worms in the foreground. Those have all the tentacular crown and are a little greenish. Those are polychaete worms. Off Costa Rica, it's been shown the sabellids can farm methanogenic bacteria. I'm sorry, methanotrophic bacteria that eat methane and turn it and use it, that energy, to fix carbon. Thank you. Good. Thank you, Tyler. So there's a high probability that all the sabellids here are doing something similar with all the methane in the water coming out in the form of bubbles. We're interested in who can use methane and how, whether the animals are getting their carbon from methane via symbionts or by eating free-living bacteria here, or by eating the other organisms. Those are all ways to get methane-derived carbon. You can see these sabellids with their big tentacles. Greg tells us it's in the genus Coney, that these are pretty common here, and they'll grow on any surface from the rocks to the clams. Good morning.
Good morning. How are you?
I'm good. We have Cindy and Alex just joining us as pilots.
So what's the plan now?
Can we zoom back out, please? We're just taking a look here before we leave this amazing bubble site. We've just taken a gas sample. Admiring this fish, which seems to be one of the only of its type here.
What do you want to look at?
I'm just scoping out what's here that we haven't looked at recently today. Is that a sea urchin? Could you zoom in straight? Have we, does anybody remember collecting a sea urchin yesterday at the Seep? No. Okay, maybe we will try to pick up this urchin, which looks quite different. Smaller spines.
You're gonna recover.
Do you think it'll fit in this section?
It's probably gonna get stuck.
You think it would get stuck?
Yeah,
Okay.
especially if there's already snails in there.
Can we put it in a coral clicker?
Yeah.
I'm trying to remember, it was either, I think it was
Well, we need to
2
find-
dives ago, we had a place with loads of sea urchins sitting on a mound. And I don't,
Yeah.
it reminded me, I've seen scenes like that of Strongylus centrotus fragilis, a small pink urchin just occurring in large masses at methane seeps off California. So urchins sometimes home in and feed on the bacteria at seeps. But we haven't seen a lot of sea urchins at this water depth, so it'll be interesting to collect it.
All empty, it looks.
Yeah.
We call these coral quivers. That's being opened now. And ideally, when there are a lot of corals, we will snip off pieces and put them in the quivers. But strangely enough, I think I saw exactly one coral at this site yesterday, on this dive site yesterday, an octocoral, and we did not collect it. So I think it's okay to use these for animals and other animals. It's not on the shell, is it? I was going to say you could pick up the shell, but I think it's just behind the shell. Oh, is it on the shell? No. The caprellid amphipods really are amazingly large here, abundant, and numerous. Yes, and strange.
No, I think you should put one underneath.
Yeah, they might drop their spines, that's the problem, and then you'll lose it.
See if that helps. That's worse. Oh, hopefully it doesn't do that.
It doesn't do it by itself, does it?
It was doing it.
It was doing it. Oh, my goodness.
So, let's see.
It does look like the shrimp are feeding on the bottom, those attached to the mud, which is interesting. Oh, come back. It's going in coral quiver 3. Great, thank you. Okay. Let's just continue to pan around just before we move anywhere. Make sure there's nobody new here that we didn't see yesterday. We definitely took a lot of push cores here yesterday. You can see our evidence of our visit. Oh, there was another urchin, so that's useful to know that there's not just one. Ooh, is that a nudibranch? Can we zoom in on that? No, that's just a sabellid and a caprellid on shell. Just curious, could we put the lasers on? How big do you think that caprellid amphipod is? This one right here, which looks a little larger than the others. There's another big one. There's a few really, really big ones here. So the lasers are 10 centimeters. I'd say this is probably 5 centimeters long.
3 or 4, yeah.
Maybe 3, 4.
Yeah.
That is really big. Most deep sea caprellid amphipods are pretty small, like a few millimeters, maybe a centimeter. So these guys are large. Oh, that's such a nice view of them. Look at that. That's worth just holding on to. Just let the video go for a few minute or so. You can see how they move. As somebody in the chat wrote, it looks a little bit like an inchworm crawling along. They are one of the stranger types of amphipods. This gives us a look at what they do and how they move. They definitely use their front claws for mobility. They don't seem to fight with each other, which is interesting. At least I don't think so. Let's see. Did you follow this guy? Like all amphipods, the caprellids will brood their young in marsupia attached basically to the stomach. You can see a few of the animals here look like they have brood pouches. And let's see, let's zoom in on one and show that. We're probably as zoomed as we can get here. And they're basically live-bearing. What emerges from the brood pouches are fully formed, small coprolid amphipods. This is true for all paracarid crustaceans, so amphipods, isopods, taneids. There's a few other groups. It also means they don't have long-lived planktonic larvae. Rather, their young just are released and crawl out and set up shop very close to the parent. You can see a lot of different sizes in this field of view, so including some babies. I don't know if the males and females have a size difference also. Definitely the ones with marsupia here look smaller, a little bit smaller than the really big one we've been following. Almost for sure they're eating bacteria, so we will be looking at the stable isotope signatures of those we collected yesterday. We have some, don't we, Paula, somewhere in I hope. Well, do we have coprolid amphipods yesterday? And I think they're in the section samples, but we should get some. Yeah. They've been a feature, unusually, of this 2,700 meter site, but I don't recall, we didn't see them at 2,200 meters at the shallower Mocha Island Seep area. Okay, that's great. One of the other things I'm really interested in is to know who's making these black patches on the sediment surface. Maybe we can zoom, not this big black patch in front of us we made by coring yesterday, but that I think we know. Oh, look, there's a maldanid polychaete. Yeah, it sort of came out, probably something we exposed. These polychaetes are called bamboo worms, is the common name, and it's pretty evident why. They have bamboo-like elongate segments. Often they live deep beneath the sediment surface, and they're typically deposit feeders. So I think the only reason we're probably seeing this worm right now is that we cored here yesterday and exposed some of the sediments. No, I think we'll be fine without it. Thank you. But it's a rare opportunity to see a unmutilated bamboo worm. As I said, they live often 5 to 10 centimeters down. Often they'll live head down and tail up. We've done experiments that have shown their tails can sweep the surface and pull fresh material down, like phytodetritus, down deep into the sediment. But in this case, the worms are probably feeding on the bacteria that are so abundant down here from as a result of the methane. It's a very nice view of a bamboo worm. Like I said, we don't usually get to see them because they're underground in the sediments. We see them when we come process the suction samples and the cores on the ship. Okay, that's great. There's also some gammarid amphipods crawling around. Okay, could we zoom in? I want to look at these dark patches, little dark blobs on the sediment, and see if there's evidence of who underground is making those, or whether that's where bubbles are coming up. Some of them have fecal casts. Excuse me. There and there. So very often there'll be like what I'm calling a fecal cast is like a sausage-shaped bit of sediment. That's the result of underground deposit feeders. And they usually will consume sediments, extract the bacteria and the organic matter off the bacteria, and then release fecal casts at the surface. So those could be sea cucumbers or burrowing anemones, polychaetes. Are we full zoom?
Yeah.
Okay. We can't go in anymore.
Try to zoom out.
And of the animal probably also aerates the young in the brood pouches. But it's sort of behind a clam siphon. It's a nice view of the siphon. These clams are getting sulfide with their foot, which protrudes deep into the sediments, which we can vouch are highly sulfidic here. And the siphon is getting oxygen. So the clams have in their gills sulfide oxidizing bacteria, most likely. The sediments are so sulfide rich because anaerobic methane oxidizers produce hydrogen sulfide. They're a combination of sulfate-reducing bacteria living with methane-oxidizing archaea, and they make all the sulfide that's supporting the ecosystem here. Of course, methane is as well. Some of the animals can use methane directly, and some use sulfide, and some use both. You can see this one clam is supporting a whole bunch of caprellid amphipods and is home to a fairly large sabellid polychaete.
It's a bolt on a.
Okay, I don't think we're going to be able to see into the brood pouch very well, so. Okay, that's great. That I've got my sort of natural history observations out of the way. It's fun to see them. We have some options, but how do you feel about heading over to this feature that the geologists want to explore? And on the way, we can try to maybe sample some orange stuff. We don't know what it is. If we see it, I'm pretty sure we will. And maybe another good Xenophyophore core. And we'll see what else we see. Okay, so we don't actually have a waypoint for this, do we, John? Do you know, did they give us a waypoint over here?
Sophia, do you know what our location is? She's pointing.
Or, I'm sorry?
She'll figure it out. Sophia will get a point.
Okay, come over. It's this, they wanted to have a look right over here. Just get a general.
I can read these out loud to you. So you wanted to say you wanted one here and one here?
Yeah.
Okay.
Should I just go right and look?
Um,
Who puts them in?
I'm putting them in right now.
Alex first.
Oh, my goodness.
All right, let me know when you're ready.
Yeah.
Okay, negative 75.
I'm sorry. Just remind me of that first.
Oh, that part, sorry. Negative 38.
Yeah.
0.46.
Yep.
81.
That's all the digits you have.
Oh, wait.
Decimals.
Negative 74,
Yep.
25,
Yep.
65, 96.
96?
Yeah.
So in mapping this area, a few nights ago, our ship mappers have created a topo map that I guess you can see on one of the, is this on the live stream also,
Yeah.
this map?
Yeah.
And so we're looking at some of the features there that we will go take a look at. Looks like a bunch of pockmarks or depressions. So sometimes methane seepage yields pockmarks.
Yep. Yep.
So what's strange is some of the things we haven't seen here at this site. We haven't seen a single tube worm. Vestimentiferan worms. These are siboglinid polychaetes. We've seen very few sponges as well, and very few corals.
Good. So you happy for us to move on?
So we'll like to just move over the bottom towards that area, and as we go, we might want to stop and sample,
No worries.
make observations. But this will be a part of the seafloor we haven't traversed.
Should be good for transit. Once you get a little, like, I'll start actually, I'll start looking at
Sophia, are you able to put the?
Yeah, she has it on that one, so.
Up there.
Looking at that one. So it's just west of where we are.
You can see the edges are fairly sharp for where you have clams and where you don't, but then they appear again fairly quickly. Usually when the clams are sitting in the sediment at an angle, which they all seem to be, you can tell they're alive. When they're lying flat horizontally, they're pretty much dead.
We've got to move into the southwest.
Yep, head west.
Thank you.
They're xenoturbellas. Could we go a little,
Yeah.
either zoom in or move a little lower, whichever is easier? Woo!
We've got auto altitude.
So the Xenophyophores that we saw yesterday definitely haven't shown up on this side yet. The Poralia, the jellyfish sitting on the seafloor there. Oh, I take it back. There are Xenophyophores here now that we're out of the seepage. We looked yesterday at some of that orange stuff on the bottom, and we weren't sure whether it was a sponge yesterday when we first saw it or some other organism, but it looks like it's amorphous orange material. Nobody's even sure if it's bacteria or if it is what they would be doing there.
We'll interpret that as you're losing things where you're coming. So I'd be careful about
But
that.
it's using.
So in general, if things are in the middle here, it's like good. You don't want things going past
We'd actually like to core like a really big Xenophyophore and also some orange material.
Just tell me when.
But I will tell you if and when we want to stop.
Actually, I think we go better fish every day.
That also looks like some kind of cusk eel.
So Urchin.
Does that fish look a little bit like the one we were seeing at the Seep?
One I see different to me.
It looks
It looks different to me.
different. Yeah. Not as round.
Yeah. The head looked different.
Does it look super washed out on this one? But that doesn't, it's not washed out over there. It's still there.
We don't know what the orange stuff is.
Okay, so those are the ones.
Interestingly, even after collecting a little bit of it yesterday, didn't seem to have any cells, obvious cells or tissues. So we decided it wasn't a sponge or a tunicate.
Yeah, honestly it is, it's fine if you go a little bit darker.
Even Greg wasn't sure what it was. Even the microbiologists weren't sure whether it could be bacterial. It almost looks like a precipitate of some sort that's non-living.
Question then, how does that impact the invertebrates or the folks in the stream relative to that? What up there looks better?
Xenophyophore.
So yeah, it's the same name,
Lisa, what has you heard Xenophyophore? Is that the name of?
Xenophyophore, yes. There were a whole lot of them back in a different
Yeah, that's way different.
area that they were sampling yesterday. We got to chance to look at the Xenophyophore in the lab. Why don't you describe, Paolo, what you thought they looked like?
Yeah, they have been famously described as a deep sea purple sock. And it was a mystery around 10 years ago, and it took scientists a couple of years to find out what they really were. But we have on board the scientist, Dr. Greg Rouse, who helped, led the efforts to identify 4 new species of them. So, yeah, it's one of the highlights of this expedition to know whether this is a new species or not. They are a very primitive species, and there has been a debate in the scientific community whether they are simplified species, so they evolved and then kind of went back, or if they have been primitive from the beginning in their lineage. That's as far as my knowledge for the Xenophyophore goes. Very interesting creatures.
Did you get to see them, Pachi?
Which is also bad if you see.
They are very slimy.
Yeah, exactly.
Here's our benthic jellyfish, the Poralia, sitting on the seafloor. Actually, could we stop here and core? This looks like there's some good orange stuff and good Xenophyophores. So I would like us to take a core over the biggest Xenophyophore you can find. So Xenophyophores, I haven't talked about them yet this morning. They're the sort of honeycomb-like creatures you see on the seafloor. They're protozoa. They are single-celled organisms within the group Foraminifera, but they're effectively giant Foraminifera. In this case, the size of your fist.
primera vez que lo vemos fuera del.
Oh, did we disturb the jellyfish? Oh, he's
Decided to leave it.
too much sediment, maybe. So this is probably a Poralia, like jellyfish, taking off, was sleeping on the seafloor, or maybe feeding. If it helps, we could try, I guess if we scooted on ahead, we'll be back into clams.
Yeah, so wait for this to darken.
Well, no, they're not that. They're not all that.
Lisa, why do we get excited for the Xenophyophores? Why do we get excited for the Xenophyophores?
They're kind of a hobby of mine. Xenophyophores are, they're interesting because they're one of the few creatures that only live in the deep sea. They're only found below 400 meters or so. They agglutinate sediments that they pick up out of the water with their pseudopods because they're basically related to amoeba, but they're multinucleate. And they create these structures. Each species has a different structure, and they act like sediment traps. So they trap organic matter, and they probably trap larvae. And as a result, they're occupied by a whole host of invertebrates, usually, that live inside them. So they're like little biodiversity hotspots in the sediment. And they also provide protection for animals under them from predators. And so the densities of invertebrates in the sediments are enhanced under the Xenophyophores as well. We know that by sampling, taking quantitative samples over them, under them, and off them to the side. And we'll find really high densities of myofauna, like nematodes and copepods inside, but also isopods, amphipods, polychaetes. We think them as a habitat, so they're like a microhabitat
That's
on the
amazing.
seafloor. The word Xenophyophore means bearer of foreign bodies in Greek. They also produce barite, which is really interesting. They have inside them, they keep their fecal pellets and turn them into building structures like bricks, but long, elongate, encased structures called stercomari. And so I think we'll just take one, a really big xenophile, like this one or this one. I don't know, the biggest one you can find. And it's okay if it doesn't all fit inside the core. We also want to, actually 2 cores, because we want to try to get the orange, a good glob of orange. And it was so amorphous yesterday, I don't think we can suction it and expect it to show up.
Okay, I'll do that.
That's all for that. Oh, I don't know.
So, number, yeah. Should be empty. Yep.
Yes, right?
Yeah.
Yeah, we
The
have
whole-
that as well.
12, and then the 13, 14, and 15 are also empty,
Okay.
as well as the ones all the way over to the right and to the left. And probably, I should have put Xenophyophores on the guide plan. Yes, I think I
Is
did.
it wrecked on that?
We'll get
No,
a
right,
good
no,
big
right.
piece. I'll show you what they look like under the scope. I wasn't sure yesterday if this is what they were, because when you zoom in on them, you see a lot of tubes, so it looks like polychaete tubes or something. But I realize there's
Yeah, if you figure out
some
the-
genera of Xenophyophores that
where the lasers
are
are.
formed of tubes. So can we find a bigger one? Let's see.
You want these ones?
How about this one?
You want-
Can you reach that?
Just so you watch my shoulders.
Yeah. Yeah, pretty good.
If anybody's interested in having the sediment under it,
Okay, what
you
about
can also
one?
have that core, but
Can't reach that here.
you can't reach that. Okay,
Así
well,
que no funciona, ¿verdad? Que
I'll look around.
let's look.
están todas las bacterias, esos chinitos, anyways, están anaranjados. Que no sería bacán.
Can you reach that one?
This one in the middle, yeah. To your right.
Is it lunch for that?
Yeah, the lasers on it right now.
Is this one bigger? I can't tell.
No, that one we were good for.
This one?
Yeah. Maybe these.
So the core diameter is 7 centimeters. I would say these are about 5 across, 5 centimeters across. It looks like he's not on it.
I'm trying to just.
Okay. There we go. Very soft sediment. I don't know if you want to push another core in to get the orange stuff or just recover this, whatever's easier for you.
Just recover this. See what it does first.
Looks like you got it. Didn't run away. So these are well oxygenated sediments. There's no black under there. So this tells us there's probably not too much methane here.
Going to go down. Is that all the way up the bridge?
So the orange material that's on the sediment in these areas where Xenophyophores are is really, really soft and amorphous. It's not rock-like at all. It could be a bacteria, like Archobacter or something that's orange. It's not clear that it is. It's definitely not filamentous. Great, thank you.
Lisa, what do you think the odds are that it's barite? The orange stuff.
I don't, does barite ever come in orange?
Yeah.
It does?
Yeah.
Well, that's interesting because barite is something that Xenophyophores produce.
Yeah, I'm just saying.
I always thought it was white, but
It can be white.
I wonder if it's what happens when they disappear or die, and there's little mounds of it or something. Is that possible? Because it's only been in the areas where these Xenophyophores occur.
Yeah, definitely.
That's an interesting thought. Hadn't thought of that. But Xenophyophores are one of the things that distinguish that group is the production of barite. Is it a lot easier to core with the starboard manipulator than the port
We have the giant ram on the working it at the moment, otherwise.
It wouldn't really make a difference, but because we have the gas light ram on it, that's making it difficult.
It makes it hard. I mean, does that mean push cores 1 through whatever? The 3 left ones are really hard, gonna be hard to
No,
use.
those are pretty much the only ones
Yeah.
we should do with that arm.
Because it's easy access to that one.
Plus,
Yeah.
there's so much stuff on the porch, we can't really come too far over with the port arm.
Hmm.
Right now.
Well, Peter, if we wanted to figure out whether the orange stuff was barite, how would we do that?
XRD would be a.
Do you guys have access to that?
Yeah, we have some of that orange stuff yesterday.
Okay, well, we're gonna collect some more orange stuff.
Amazing.
So we're gonna look for a big patch of orange. Maybe you've got one there. That looks like a great spot.
Yeah.
Yeah.
Okay. Go for it.
The ocean has, deep ocean has so many mysteries, and we come across things that we don't know what they are and can't explain. It's one of the things that keeps scientists going through these long hours. Most of the people on this watch were up very late last night processing samples from the last ROV dive. Same with the watch before, actually. Certainly in Aaron's case and Maritz's case. I do see like the Xenophyophore behind us in this view has some orange stuff next to it. Whatever that's worth. It's very amorphous, and as you can see, it looks like it's in there in the core, but not holding a specific form.
Right, coming out before.
Once he gets it seated in the quiver, you could zoom in on the top of the core, and we can just see what's in there. This is the second to last dive in this leg of the expedition. We're hoping to have another dive at 400 meters closer to Mocha Island at about 8 p.m. tonight. And that will be our last one. It'll be a short. There's a polychaete released. There's definitely some orange in there.
Sorry.
Here's some discussion on the chat about the newspaper we saw yesterday. It was about the Olympics, so it was clearly a few months old, but newspapers are probably made from wood, and wood has a lot of cellulose. It's hard to digest. There's not a lot of different bacteria that digest it, so the fact that it was intact at these cold temperatures and high pressures probably just mean there's limited microbial activity down here, and the animals don't eat it. That's great, thank you. We got some, but it sort of falls apart quickly, so we'll see. We'll send you home with some more Peter for X-ray diffraction. That would be so cool if it was buried.
That would be neat. Yeah.
We've seen surprisingly little wood down here off the coast of Chile. Greg and I were remarking on that. Okay, we can continue on our trek. Thank you. How far is it? Like 100 meters or something like that to the first waypoint? 885? Yeah, thanks. Xenophyophores normally don't occur around methane seeps, so my guess is that there's plenty of oxygen and very little hydrogen sulfide right here. But interestingly enough, there are patches of clams not too far from them, so it suggests the sulfide's really localized. You can see these little clusters of clams, purple sea cucumber. Oop, a peniagony maybe, swimming just above the bottom. We collected a beautiful one of these yesterday. That's a nice view. Let's take some video of this fellow. Pelagic sea cucumber. It's nice lighting. Got a BBC. I'm going to zoom. I don't know if zooming will work. They've clearly been down on the bottom deposit feeding. You can see their gut is filled with sediment. One of the pictures I got of the Anipniastes at the very beginning of the cruise has already appeared in a policy brief, Dosi policy brief.
Amazing.
It's like the perfect position. That's a nice photo. Okay, we can continue. Thank you. Nice pictures. Video. What's amazed me is how really large this whole area is. We've seen seep patches sporadically, pretty much everywhere we went yesterday and today. But we'll see what's out here. There's no guarantee that there's methane seepage where we're headed. We're moving toward sort of a almost pockmark-like area. It's been mapped, but nobody's seen it. There are definitely a lot of Xenophyophores down here, really abundant. Be interesting to know what it is they're feeding on that's got them here. Can you zoom in on this? It looks like a bowl.
Oh.
It might be a bowl, like physically, a human bowl. bolopathies. Unfortunately, more evidence of humanity
Yes, I know.
left for explorers thousands of years from now will find our debris and try to piece together our civilization
Mira, sí, ¿verdad, chiquitito? And this is really beautiful.
from the orange bowl.
That's right.
What's inside? There's clearly a worm tube, a couple of worm tubes in there.
Yeah.
Wonder if we could figure out how long the bowl's been there based on sedimentation rates. It looks like might be a terabellid polychaete. Hard substrates get used by animals, although I'm surprised there's not much living on the outside, on the surface here, and it's probably plastic. Plastic doesn't degrade very fast, so it's a real problem in the deep sea. It comes in many forms. Here's a big piece of plastic, but there's also microplastics down there that all of the animals end up eating. There's a plastics treaty being negotiated internationally right now that's covering the full life cycle of plastics from production to waste disposal and everything in between. It is very contentious. Okay, good bowl images. Ooh, that Paralia looks like it's got eggs or parasites. I don't know what's going on with that one. Do we see that? No, it's not a Paralia. It is one of those big Plurobranchias, I think, that we saw the other day. Let's get a good video of it. These are sea slugs, giant sea slugs,
Okay, yeah, go ahead.
which are shellless mollusks. I forget if we collected one. I think we did, didn't we, yesterday?
I don't think
They collected it.
Do you remember?
Unless we get a request for it, I'm not gonna take it. But we definitely can get some pictures. First I thought it was a Paralia on the bottom, but oh.
She's moving quite fast, isn't it?
For a snail,
For a
for a
snail.
slug, for a slug, yeah. Not really very fast.
Faster than deep
A
sea.
centimeter a second or less. It's beautiful. Not sure if those are reproductive structures on the back or something else. It looks like a nudibranch. It's related to nudibranch in that it's a mollusk, but apparently it's a plurobranch.
Lisa, can we hold that for like 2 minutes just so that I can
Yeah, you want to
run
back
time
out?
lapse?
Yep. But can we back, you want to back off of it, or?
No, 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 it's good.
You like this view? Ooh, look at all the little fecal pellets down on the bottom, right outside the worm tube.
Lasers on and off.
It's so fascinating how, as the mollusk moves, it's totally not disturbing anything that it's crawling over. So those xenophyophores are really fragile. All those fecal pellets haven't moved a bit. That's really interesting to see. So I wonder what it's eating. So you don't want us to
He's doing
back
it.
out on it?
No, he's doing it with his
He's
camera.
controlling it. That's great. Then we get a good view of the orange gunk.
Every one of those is a single cell then, is that right?
It's a multinucleate single cell. Oh, there's another snail. That's a snail with a shell.
So it's all sort of just sloshing around in there. Lots of nuclear
He's chasing around.
This one?
I'm joking.
They might be
Yeah.
snail predators.
What do you think are the evolutionary pressures that drive an organism to be multinucleated?
The absence of cell walls.
Not sure.
We don't know. I mean, xenophyophores are really big foraminifera, basically. People thought they were their own group, and then they sequenced them and found out.
Crawl towards us.
Potentially, yeah. Just hold that.
So
Hold that
getting
for a
big
second.
is
Yeah.
probably driving the multinucleate part. I'm sorry.
The battle of the slowest entre Snail aquí y Urchin.
It is a sense.
It's so amazing, though, how they don't disturb anything on the bottom at
It's
all.
nearly-
It's going to eat it?
I don't know. He's gaining on it.
Where is
He's still, you were seeing it, but he's going over it.
He's over it now?
The snail just disappears.
Yeah.
If he comes off the other end and there's no more snail,
Yeah,
I
the
guess he
snail
ate it.
just got, disappeared right around here. So he keeps
Interesting.
going, so you don't
Well.
see the snail. It was gobbled up.
That will help solve the problem of what they're eating. Or if there's an empty shell that comes out the other end.
Sí, yo creo que sí. Lo que será fue.
Yeah.
So for those on the chat, the orange stuff is not a sponge. It has no
No.
cells as far as we can tell.
Snail.
Oh, there's the snail. So it wasn't a snail predator.
Ay, Sophie.
Snail looks pretty intact. We don't know what the orange stuff is. Current theory is it could be barite, something produced by the xenophyophores. It could be bacterial, but it didn't look like there were any cells in it. Looks like an amorphous precipitate when it's collected. Doesn't have much structure. Do we want the snail? Okay, so we have a request to suction the small snail, not the big slug.
See hands on it?
Yeah, you can let them be afraid of them. Which is
Our
good.
frame or your-
Sorry?
Our
You're
frame.
afraid.
I haven't been following the chat.
The slowest eating frame.
Is it still on prep?
It's
It could be into 5 or 6. Did you hear the section?
Yeah.
So what are we doing?
We're just waiting for this Snail to get out a shot for BBC. He's waiting for the.
Oh, he's waiting for it.
Yeah.
Sorry, yeah.
Oh, it's okay. That's fine.
No, we're-
Push him.
Other seconds.
We're all poised this section.
Any second.
We don't want that in the video.
Consider it a sort of mindful moment. Just watch the snail. Really great.
Good morning. We're doing some BBC snailing.
Nice blue little black. It's a bestial.
The big one.
Flat musk.
That fits up in suction sampler. Too big for the suction sampler? That's fine.
No, we are not suctioning.
Sorry, I'm putting ideas in
Put him in the Bible.
You would fit, but you can leave the poor guy alone.
I'm joking.
Okay, there he goes.
the sunset, isn't it?
Hopefully green flash.
Is that okay? Is he off enough?
Yeah, that, yeah.
On the
All
shadow
right.
though, do you have a shadow?
That's all right. Very good shadow. Sweet, string bit. It's nice, it's strange. Working great.
Yeah.
What?
I have to put the lid back on.
Okay. Alright, I'll fly forward.
Might actually need that out for the bungee to reach. If you're trying to, oh no, the bungee's gonna make it.
Alright. Bring it on, let me file.
Is that too far? He's gonna ask you.
Yeah, you can, obviously, about it.
What, number
Which one do you
7?
want?
Not 7, because it's filled already.
So.
Can you index
Eight?
to, no, 8,
Six or five?
6 or 5?
Cool.
Okay. 5 is fine. So you're just getting this small snail. Did Javier request that?
Yeah.
If you want to,
There's two of them. Right here.
oh yeah, 2 would be good. Well, also, I don't mind taking a piece of Xenophyophore. I don't know how it'll go through the tube. But
Suction is on.
So we're already two snails stuck. So, like,
Yep, there's number 1.
where's the second?
Right in front of number 10.
Right down here.
Okay, it's in.
Yep, got it. That's good. Yeah, great.
What else you said you wanted? This one?
Um,
Okay, those are the ones. Index.
I think that's fine. So that was section 5.
Thank you.
Yeah.
Go 6.
It was 6?
Sorry, 5.
It was 5.
Thank you.
Check on it.
Well, we're almost at those features that we're supposed to check out. Get over there soon. We just keep running into cool creatures. Well, that large pluribranch might end up as a Liffey star. You never know. Now that it's been filmed by the BBC. Giant Chilean sea slug.
Sí, una botella. Oh, another garbage. Garbage. Yeah. Ya lo marqué.
And
Bye bye. Beautiful. Little horns.
Qué chulería.
Is that our skid marks?
Yeah.
Okay, so we're approaching the area we're supposed to be checking out. It's showing up as kind of pocked, rough topography. I'll have to see what it is. I guess we're not quite there yet.
No.
Oh, big anemone. Haven't seen those in a while. Beautiful. Sea star. A ledge of some sort.
Okay, we're about to retrieve one.
Look it up.
Oh, there they are, depressions. So this is, see how it
Yeah.
goes down maybe half a meter or a meter? Sometimes methane can cause depressions like this, little pock marks on the seafloor. Eel pout, maybe. What do we think this is? Could that be? Wouldn't be a toothfish. I don't know my fishes. It's not a chimera. We're on new terrain from yesterday. We've come over to a feature that looks like a whole bunch of really big pock marks. We're checking out what's here. Ooh, could we zoom in on this to see what it is? Something new and red. Looks like some kind of pelagic cucumber, but not one I've seen. Not even sure it's a cucumber. It's beautiful. Might be a cucumber. Not sure what else it would be. Interesting color. Greg said a Xenophyophore. So, interesting looking Xenophyophore. More debris.
Uh-oh, you're going to circle death.
Not sure what that is. Something rusting.
There's one vessel,
Ready, Ruby?
Yeah, go ahead.
Can we move you south of Blue Side?
Call down.
Thank you.
you want to look at this?
No. Unfortunately, just, we documented it's there.
Thank you.
You know what's funny about this?
You were paying
Yeah. That was worth a try.
There's another
Yeah.
pelagic cucumber on the seafloor.
Yeah, we'll go away.
Sonny, you're in the circle of death.
No worries, no worries.
Sonny,
You're only 3,000 meters now. Not like when
Well, I would say this terrain is more of the same, just a little bumpier.
Interesting. I didn't know that. I just thought red.
Like an eel pout.
Because my perception. My perception is you go in there and you're
Yeah, like birds.
Yeah, there's some zoom on this fish.
Sure. Could it be a snail fish? I'm not sure. Elodio wants the logfish. That would be the eel pout, I think, that we saw on the bottom. Were you able to section that?
The fish?
The long one. He said not that little one.
If I see one, then I'll try to. Okay. The city fish or so, it's not the long one.
The eel pout.
What kind of fish? If we see one, then we need to.
I just asked.
Yeah.
We'll try to find it again. The eel pout.
But I'll research about that, so.
Does he know that?
Yeah, they do. Once they feel that current, they just can't help themselves.
We just need to bring harpoon next time.
Does he know that? I know, but like,
ese tránsito para Chile, Mocha Island. Después, bien turista, ¿verdad? Es que vi que tenía un hotel. Voy a preguntarle.
Ooh, what's some white stuff on the bottom?
Do we know if we're in the transit for tourism for Mocha Island, and that's why we're seeing anthropogenic debris?
I don't know whether there's tourism. Maybe Pachi knows.
The Apache said there's not that much for Mocha Island, but they have an hotel.
No fish, huh?
Bueno,
Ah, pero había una como que
coming down on that, like,
representa-
usually.
Sí, porque es que cuando estábamos haciendo chic, de como que Airbnb, estamos diciendo como que
Could be a skeleton of some sort. Something dead.
no muy fancy.
Okay, continue.
Eso parece.
Look like some fish bones down there. Well, let's zoom in and see if there's any ossidacs, which are the bone worms. Looks like there's a lot of amphipods and isopods on the skeleton. Looks like fish vertebrae and
I'm not moving, you're moving, right? I'm moving.
don't see any bone worms, ossidacs.
Alright, presume.
Definitely some good sized isopods and amphipods down there. Scavenging. When something dies and falls to the seafloor, it attracts a whole bunch of other animals. Looks like there's a scale worm on it. It's the closest thing we've seen to a whale fall, but this clearly is not a whale. A fish fall?
Because it's on my list to see a
But what are these weirdo structures? Does this seem like it would be a skate arm or something like that?
Jaw of like a redfish.
A jaw?
Lower.
Oh, the lower jaw of a big
Looks like
fish.
a whale.
Just going off of like
Yeah.
experiences with like redfishes.
Uh-huh.
Redfish, snapper. That's what you as the underside of the lower jaw you have.
Let me see if Greg has any interest in this. Okay, he's not interested, no ossidacs, but it is really nice to document all the amphipods and other creatures that are using this habitat. Again, like the dead animals are a microhabitat on the seafloor that help promote biodiversity. Different creatures use different little environments. Nice view of them. Well, Eulogio's interested in suctioning the amphipods.
Yeah.
This. Yeah.
Some, you'll get some bones as well. Right?
The bone is fine.
Yeah.
We don't mind.
Alright.
Um,
Suction time.
Like one of those.
What,
let's use number 4.
would you reach that or do you need to do it closer?
Bone bugs, that's a nice name for them.
When we had some of these.
I know, but it's really good. Yeah, so many missing stuff,
It's actually surprising how few skeletons we have seen down here. I think this might be the first one.
So we're trying to get the bugs or the bones?
Oh, well, basically the bugs on the bones, but
The bones might be bonus.
but if we get the bones, it's fine. You can see they're small compared to the size of the
Yeah.
suction hose.
Start turning this on.
So I'm concerned if we get the bones, they're gonna clog the line.
Amphipods don't want to let go.
They get stuck. So we might look at this.
Oh, snail came in.
Please, not a little bit.
Yeah.
Oh, the snail from before?
No.
Or a different snail? That's a different snail. Where'd that come from? Maybe it was on the bone?
Well, hopefully the bone makes it through.
Shake it.
Let me make sure the hose is clear.
I mean, the bone wasn't so large.
It was big enough to be stuck.
Hmm.
Take it out to the right, like, shake it higher, yeah. Yeah, shake it.
Is the suction on while you're shaking and
Yeah.
it'll should move it. I don't see any amphipods going in either.
You wanna, just suck some with it?
I don't know if that's it. Did you turn the suction off and back on?
Yeah. Yeah, just skim a little bit.
I don't know if this particular button is super useful.
Okay.
Like all those little critters.
Maybe critters are in now.
It's good.
So is the bone.
You see the bone? Come.
Okay, shake that out for me, okay?
Definitely some amphipods in there.
Yeah. So they got no loose.
Just one more back and forth. Bone's probably stuck at the transition
Into this canister.
well, where it goes, yeah, where it goes from hose to right after. That's right. I'll get that
that's fine. We got some.
Alright.
So that was section 4. So we've pretty much come to the second waypoint, right?
Yeah.
Okay, let us, oh wait. Let this clear. I would say we could go just a little bit farther and then turn around and head north to, what is this waypoint called up here? We haven't explored the feature of Todd. Yeah. So let's continue west just a tiny bit further.
Okay.
And then head up to that one. So basically the features that people wanted to look at over here is just some rolling topography, but not a big change in biota and no seepage that we can tell. Another sea cucumber. Sand home, empty. Lots of orange stuff. There's some brittle stars under. Do I you want to take another push score? Or was that a joke?
I don't mind. I don't mind.
All right, why don't we grab more orange stuff in a push core? Since we have a lot of extra push cores, let's get a really, really big, thick patch of it. Okay. Peter, are you good with that? Get a really big chunk of this stuff?
Yeah, I'm
You don't mind opening the cores for us?
No, I got you.
It's just such a mystery because there's so much of it here.
Yeah, well, and you pointed to the Xenos as a likely source and
A possible
a
source.
possible source.
There's some large marine snow goo. Maybe from a larvacean. It's just very expensive, and I've never quite seen anything like that on the seafloor before.
There is Seep associated barite, but sometimes they form like even barite chimneys. I've never seen it like this.
I've seen it, but the barite I've seen is Well, I've seen barite form around DDT barrels
Oh.
when caustic contents comes out of the barrel, something alkaline, and it reacts with the sediment and makes an orange halo, like around that we thought was bacteria, but turned out to be barite, or at least partly barite. So I've seen that, but the barite at Seeps I've seen is white, which is why I was asking.
Yeah, so, at least at Point Leon, inside the chimneys, there were orangish layers that were, at least by XRD and, electron dispersive spectroscopy, they look like barite. Now.
You should talk into the microphone so everybody can hear.
Sorry.
Hear your wisdom.
Want me to adjust it for you?
Is this good? Sorry, we're having a heated debate on the identity of this mysterious orange substance at the bottom of the ocean. Now, at least some of our microscopy observations on the ship don't indicate any obvious cell structures, which leads to the potential for mineralogic origin. Now, there aren't many minerals that have this bright orange feature. Barite is one of them, but as Lisa has pointed out, many times at methane seeps or methane-associated environments, it forms a more of a white color and a texture that's a little bit different from what we're seeing today. Now, that being said, there are examples of this orangish color, so if you look up orange barite, you'll find plenty of examples, but it is a bit unusual to see it in this form here. That being said, this is, as I understand, a relatively unexplored region of the ocean, so it's very exciting.
I mean, it may not be barite at all.
Yeah, it could be
something else.
Yeah.
There's literally no
Well,
What did she say?
she said there's literally no way of knowing. Is there might be, let's see, so it's barite is a barium sulfate, so if we were able to have some sort of reaction to, okay, this is very hypothetical, but if we had something on board to react with the sulfate, but I don't know what we have.
That's touchy. But we have.
So cool.
You think, so if we had like a sulfate mineral, what could we do to try to prove that without like XRD or something like that? Is there like a substance we could add to it? If we add water to it, it could dissolve, but I think we would have to. Yeah, because sulfate.
You can see it by microscope.
Yeah.
Small rings, black rings that probably correspond to
Okay.
theory.
Yeah. Yeah, that makes sense.
Greg is sending us pictures of the orange material. I wonder. Where did the big orange blobs go? It's there. The mystery of the orange blob. that was good. Whatever you just did.
Well, it's current. This week we got almost.
This job requires a lot of patience, doesn't it?
It does.
Do they give you a patience test when you apply?
Let's see.
Yeah.
Lo llamas así cuando es como una plancha. Como que no muy fuerte.
Where are you from, Zoom?
Trinidad.
Trinidad. A good friend from there.
Yeah.
Well, you've met her, probably, Diva.
No, I've heard about that.
You've heard of her. You never met her? Hasn't she been on board when you've been on here?
This is my second trip on, so I was first time I was on here was June this year.
I see. She was on earlier. Yeah. Got it. Don't run away. Did you ever meet Judy Gobin?
Next one up, I know the name, but I'm
You
not.
know, yeah.
I'm-
Well, these are awesome ladies. You'll meet them someday.
Yeah, I'm new to
Deep sea science.
exploration and science. In coastal oil and gas before. Our paths met across here.
Nice. All right, well, if that's not enough for us, then we don't deserve to know what it is. So we have to figure it out from this. This might be our deepest spot on in this area so far, 2758 meters. Great, thank you. Okay, why don't we head up towards the waypoint in the north, and we'll go exploring to the north of that even. I think we've been there before, but not all the way up.
And if we see that eel fish, we want to sample of
Yes,
it.
we do want to section the eel pout if we see it. So much orange stuff. So many Xenophyophores. So hopefully we're going to cover a little ground that we haven't before. You can see it's uneven. It's unclear what would be causing the mounds here, unless there's, I don't think there's methane underneath, or we'd be seeing clam shells and other signs of seepage. I don't know if they could be sand waves. Little brownish mounds are Xenophyophores, as best as we know. Xenophyophores have names like Reticulamina and Galatheamina. I need to look up which one is more tubular in nature. That is not the fish he wanted. Looks like a snail fish. Liparidae. That one might be. This eel
You
pout.
might have a target.
I hope there's no bones in the way. Look at the fish got on the bones in the hose. Pretty sure that's what Elohio wanted, a little eel pout. So there's a discussion going on in the control room about whether the orange spots could be barite. We know that Xenophyophores precipitate barite. It's one of their unique features, and so we were talking about how could we tell whether the orange material was barite, and is there a acid that could be used to test for barium sulfate. So Eulohio requested a suction sample of the eel pout. We're trying to get enough visibility to sample one that we just found.
We can apply this one.
Ooh, is this a ledge of some sort, or
No,
just
no,
just
you hold
a?
on to it, just keep moving.
So if we have the HCl, we see no dissolution. And we have the HCl and the nitrogen, we see dissolution. That indicate that
But
they're-
still, is this a ledge of sediment?
No, no.
Or it's just the image?
Has
It's just,
the cloud.
yeah, the cloud edge.
5 meters up right now.
Okay.
Otherwise, we would shoot X-rays right there.
Yeah, that's good.
It's getting worse. What's the deal? Is the current coming at us, or what are the chances that fish is still going to be there?
Keep moving, and if we see another one, try. I'll continue on to the Let's keep peeking down there. Yeah.
Where's the test back?
Ooh, there's another one of these egg cases. We saw one yesterday. Decided it was probably a large snail, some kind of mollusk. Ooh, could it be the plurobranch? Do that? They're the only really big mollusk we've seen. It's the big sea slug we were just filming. I wonder if they make egg cases like this. We collected one yesterday, so we don't need any more. But They are massive eggs.
Yeah, go ahead, man. We gotta head away back to the. All done.
Ooh, clam shells. We do know the northern area we're headed to has got seep features, lots of clams and things. Wasn't fully explored yesterday. Oop. This is the guy I was wondering if it could be the egg case of this, because it's the only really, really big mollusk that we've seen. And those eggs definitely look like gastropod or mollusk eggs. Here's an acroax shell. Acroax is a type of clam that has symbionts in its gill, like the vesicomyids. It's found at methane seeps in various places. And they're large, but we only found dead shells yesterday, is my understanding. I think they're solamyid clams. This looks human, like human debris, but maybe it's not. Maybe it's a sea cucumber of some sort. Yep, it's a cucumber. Greg can tell us what it is. We're changing the watch now, so I'm signing off for this dive. I think the required cores are taken. The gas samplers done. Got some orange blood. And this is what's in the section sample, a variety of miscellaneous creatures. Snails, a green cucumber. I wasn't here for the worm. Amphipods. So we are headed, they wanted to explore this sort of bumpy area, which is what we've been doing. See where we are, we're headed to the northernmost wave. That dive went about half, yesterday we got halfway out that northern feature, so we were
Right.
Oh,
You want to sit here? You can use that one.
Well, good morning, everybody, from off Isla Mocha in Chile. We are back at the same site as yesterday for our final dive here. Continuing to explore a 2,700 metre deep seep site. and we've got a couple more hours before we have to leave the bottom. So we're heading to a part that was mapped yesterday evening by the at the end of the dive, and it's got some interesting topology. So we're going to head over there and have a look. So you know what we've done. That's what we've done. So there's a lot of brown encased shells you can see there are called aggregations. And we have not been able to collect one alive. We keep getting the dead shells when we think we've caught a live one. So that's a bit of a missing sample in our list. The other shells are big vesicomyids. We have successfully collected those. Not on our list for today. I am after pink sea urchin.
Okay.
This is one of those we'll get.
You just point them out and we'll capture them.
So we had an early start to dive. Our early start to this dive, the ROV was in the water at 3, and quite a lot of sampling's already been done. And I'll be narrating this morning Greg Rouse from San Diego, along with Elohio Soto, our chief scientist. Do you want to introduce, talk? Here's the microphone.
Buenos días, nos encontramos investigando frente a la isla Mocha, 249 metros.
You want to post that? Kind of crabbing along at a kind of gradient here right now.
Sorry.
I'm crabbing along like a gradient. That's why I'm kind of going sideways instead of like, go that way. You wouldn't see anything.
Did you say something? Yeah.
Okay, that's me on the marker there, almost.
So we came around the edge of this?
Yeah.
Yeah. Well, let's keep going to this. Do we have a marker?
So the white line is us.
Yeah.
And going up that way.
Yeah, let's have a look around here, and then we'll probably go back and look over there.
Okay. One of you right in front of us.
Yeah, we can select that.
Look.
That could go into 8. Please.
So 8 we have,
Oh, you've already done 8?
yeah, we've got one of them in it. We actually grabbed one on the way down.
Oh, awesome. The one that looks like that?
I believe so, yeah.
Or the big...
Oh, I couldn't tell the difference.
Oh, okay.
But we still have 6 open.
We could put it into 8 as well. Was it near the bottom?
Yeah.
It could be the same.
3 meters from the bottom.
There it is.
You want to bring the thing out in front?
Well, we need to index over to the jars.
Oh, yeah. Which jar you wanted then?
It can go back into 8. Ooh, that's a floater. Fantastic.
Where is it?
That's the big one that I want to tick.
Where is it?
Ah, no.
No, we don't worry about it. You got the main one. Fantastic. If we see another one, we might stop it. But maybe index off 8, because it's pretty floaty. See, it's going back up.
Suction, don't
It's going up.
index yet, suction.
It's going up into the hose.
Yeah, it would just get chopped up if there's
Yeah,
index.
yeah. Okay, so index off to
Yeah,
the sea cucumber.
to 6.
Yeah.
Go to 6. Or between is perfect too. Yeah, either way. That's good.
So we got a worm in 8. We got a worm in 8.
Yes, they got it on the way down.
On the way down. It's on the way down.
Sonny, do you have any idea when you got that worm on 8?
It says 8:14 on this, at 2,700 meters.
8:13.
Whatever time that means.
No, it's probably closer to 5.
4 hours ago?
It's right when we got off, before we
Just-
saw the seabed.
Just when you got to the bottom. Great.
Somebody must have time with Mars.
I'd ask the pilots to keep an eye out for some pelagic worms, and one of my favorites is called Floater, F-L-O-T-A, and they got one this morning. We'll have to go back and look at just as they arrived near the bottom, like 4 hours ago, nearly 5 hours ago. But now we're here looking for seep activity, and we're hoping for some rocky substrate. bottom's got lots of convolutions here, mounds and little valleys, and we're just going to look down in some of those. I think if we go kind of down in here,
Mm-hmm.
check that out.
Yeah, no, the direction I'm looking at.
Yeah. One of the striking differences from the other day, if you remember our dive at 2,200 meters, we had a lot of. Oh, look at this. Yeah. So let's look along this edge first. There's an interesting map going on.
I already messaged her.
I'm a bit concerned about trying to core into a slope like that.
I'm sure everyone's concerned.
What?
I'm sure we're all
So,
concerned.
have you ever cored into like a slope like that? Does that
Well,
work?
we do have
You
that.
can core to a slope, unless you're worried about slope stability.
We have the upper edge.
Yeah, I just came inside to show you the.
Yeah, we'll just look along it, and then you can make the call.
Okay.
Because we have plenty of calls.
Yep.
We've got 1, 2, 3, 4, 5, 6, 7, 8. Oh.
Yeah, could we get a bit of a zoom just to see what's alive at the top?
Could you do the zoom there, Alex?
What's that?
Just zoom, pretty color.
I'm just trying to see what these little white things are. Oh, there's snails. Okay.
Zoom on.
Yeah, zoom out's fine.
Just bring it a little closer to us and kind of like in your little bag.
Yeah, that's. I was trying to zoom it further because I was assuming we're just going to write on this stuff. Pretty good, Jeremy.
Go ahead.
You got a innovative point.
Up there we go.
Thank you.
Yeah, that spot up there looks pretty flat. It looks like a nice map. I think we could go there.
I don't know.
And we're after 3 tightly grouped.
Okay, what I think we'll do is go and sit up here facing the edge, and we'll do some coring and probably some sloping. That'd be great.
location,
Yeah, that looks good. Yeah,
Maybe.
probably if we can core in here, would be great. Nice landing.
Push past it, it's not push right past it. No, I wanna go too close to it.
Could we do a little bit of zoom before we push anything up?
Yeah.
Just get a picture of the mat and
Greg, there's a large ball shape in the aft camera. Is that some, can you see it?
sorry, which camera?
Do you know what it is?
Oh yeah, that's a big mollusk.
Mollusk? Wow.
It's a big, it's called a side gill slug. And I think we think that's what laid the big egg masses that we've seen in a few. The giant ribbon, we did collect one.
And what is this?
That shell is called, it's a clam called aggregations. But we've got some cool little snails and there's skeleton shrimps hanging out. I think skeleton shrimps yesterday were one of the features of the dive. Unusual to see them in this setting. And so many, and they're quite large. They're a couple of centimeters tall. Had fun photographing some of these last night. Normally we see them hanging on seaweed or bryozoans. They're preying on plankton. Who knows what these ones are eating? That'll be something we'll try and find out. Got quite a lot of tubes coming out as well as the clams. Yeah, so that coily tube there is some kind of frenulate. So that's a relative of the lamellobrachia tube worms that we've been seeing. It'll be tricky to sample. Could we look at these tubes over here, please? Yeah, so this is looking like a pretty classic seep mat with lots of small gastropod snails and polychaete tubes. And we're going to take some push cores for microbiology and then we'll probably do a bit of a slow. So, Peter, do you have a place you'd like?
There is no Peter. There is Amanda.
Oh.
Sorry.
Well done. We found a
I
mat.
know the long one there is.
Can we pull back? Oh, you're just fixing that.
I just put section 7 away. No, but just in case there was an opportunity to fly over. And then we're gonna do push course in this map.
So we just saw this mat on the edge of this. We're on a slope right over here. It just drops away.
Yeah.
We just saw.
the side, or is it extended to the top?
We're at the top of it, and it goes down steeply. So there was no mat anywhere else. I mean, you've already got mat from arrows, but there's more calls available. So figured you might as well use them. Peter suggested 3 tightly grouped. Do you want white mat or black mat?
Black mat.
I'm thinking, so like kind of here?
Yeah. Well, that's on the slopes. What up? Here where it's a little bit flatter. But where, like
I
shallow.
think it's straight right out. It's flat right in front of us in this view.
Is that flat?
This is flat.
Or on a slope?
Well, the slope is over the edge there. We're flat right where we are.
Oh, okay. Yeah, because it looks like this. It does look like it's not it.
It's-
Wherever it's flat and in a mat, it's fine.
Like
Whatever
here?
works for Sunny Dover. I would like the dog. Probably much flat.
Sorry, Amanda, you come here and look with me, please. Could we zoom in here, thanks? And then down to the blackish. So,
In there looks great.
so where,
In that
that
strip.
kind of stuff there, or up here?
So avoiding this.
Yep.
But trying to aim more for patches.
Okay,
like
and we'll
that.
do 3 close together. Okay, all right.
If possible. Thank you.
Okay, Doug, so we want to not get it in the brown, but in the grey or this kind of grey area, 3 fairly close together.
You can keep it
Starboard
repeated.
around.
This
Skin
one?
bottle before. Thanks.
Okay,
Take,
we'll do a Niskin first, please.
this one.
Swap out the tile.
Yeah, I'll just hand over the towel. Give me something I need.
Thanks, Sonam.
What's that mean?
Right, all right.
Alright, golf disc number 3. Retract here. Yeah.
Sure say hi.
Yeah. Definitely. Alright.
Great.
This is sense.
spot, right, Greg?
In the gray, yes, please. Wow, look at this. Some great. That looks pretty good. I think, Doug, if you do them in a line, going along the grey that way, would be great. Thanks.
Right here is good?
Yes, please.
Okay.
Okay, great. tubes coming out of the bottom called frenulets. So it's related to lamellobrachia. So if somehow you get those tubes, would be good for me.
Great.
Wow, look at all the amphipods in the core.
No charge for this.
Huh.
Now for the fun one.
Yeah, that looked a little interesting.
Amazing, thank you.
So, now we'd like a bit of a sloping, please.
Here?
Yes, please.
Okay, keep it out.
Sorry.
False alarm.
Yep. And we'd like the surface, and there's lots of little snails and the mat itself and the little tubes. Might have to dig in a little for the tubes. And we could do this into 3.
Got it.
Where are we looking?
Doug, if you see this sort of line along there, there's a whole lot of little shells
Okay.
just running along there.
Right around here, yeah.
And then just past where you caught.
Section coming on.
Yep. So those are classic seep snails called Provana. There's some little vesicomyids in there. So mostly they're, oh yeah. Mostly those are bivalves. We saw some actual snails somewhere. Would be good to get those.
I think we saw before on the top.
This one here looks like it might be a snail.
Yep. Yeah, that one might be a dead one.
That one.
Oh yeah, there's a snail. Yeah, that is a snail.
Oh, that's
Might have been a dead one.
Yeah.
Sorry, let's have a look back a little bit. Near where you can reach.
Can you name the tubes again, Greg?
Frenulate. How about just past the coring? Can you reach that far? And we'll zoom in and have a look.
Just over here.
Yeah, can we have a look at what is there? Oh yeah, they're live snails. All the little white ones. That looks like something we call Hyalogyrina. Nice. Oh, and some big polychaetes in the tubes there. Whoa, it's a lot. You could dig it in a little to get those. Thanks, Doug. A couple, yeah. That's great. And, oh yeah, juicy. Tyler, can we just zoom on that? Those. Yeah, so the white shells are very common on bacterial mats. Beautiful, glassy, translucent shells. Hyalo means glass-like, and Gyrina is the rest of the genus name. Can you do more around this area? There's a few more of the little shells and a few worms out on the surface. Those are probably what we call capitellas, the red worms. Okay. There was a brown snail that I did see. I'll just see if there's any of those nearby. It's like we've got the nice white glassy shell, so there's a brown snail somewhere. I thought these were it, but they turned out to be bivalves. Can we look back just there? No. Okay, that's good. Thanks, Doug. We'll call that. Thank you. Yeah, could we just go and have a look at the drop down and then we'll probably head back south. Yeah, thanks.
What's that? Sweet. Alright. Right.
Yeah, because it also
Yeah, it's up there, so yeah, it's not very. Anything specific you want to look at down
Yeah, can we just look at this little mound here? See what this white creature is there. Oh, it's a Xenophyophore. Okay, the stars of yesterday. And one of the stars of yesterday. Okay, what else have we got down here? Kind of on the lookout for rocks is why we came over here. I'm guessing it's pretty soft bottom and not a lot of obvious seep animals. I think we could head back the way we came, Tyler, and we came over this feature. We came around that way. So I think we came around the west edge of it.
where they came from.
Yeah, so maybe we'll go on the east side of it.
Okay. Over here,
Yeah.
towards this M3
That-
POI.
That's, yeah, that'd be good.
Red RV, got an updated waypoint.
Copy that.
So we've come north of where we were yesterday. Yep. All good? And yesterday was very much dominated by large clams further south, very active. And the mapping node revealed this more varied topology. Is it trash? Yeah. So we've come up here. We're heading south again now. There's still a feature south of us that we came around one side, we'll go around the other, see if any rocks are there. Another newspaper.
It's not the same.
Different day.
Yeah, it's the same newspaper as yesterday.
Yesterday, this is.
Oh, it's the same, we came over this?
Yeah,
Ah.
that's where the ROV sat now, right there. That
Oh, I
little
see.
skid mark.
Right, yeah. That was a page from back in August, in the Olympics. September. And we've got some large white sea cucumbers, Panayagony or Actimeria, large jellies, probably Paralia. Saw a lot of those on the bottom yesterday. And there's somewhere else we sat down yesterday.
I seem to remember the area we were headed now. It was full of rocks yesterday. So hopefully we'll find some. It's clipped.
Yeah.
Because I remember last time, like. But now you know.
Can we have a look at this, please? that's all feather duster worms. Yeah, it'd be good to get some nice video of this. Great. So they're all called sabellids. And there was another nice field of them up over here. Thanks, Tyler. Yeah, just if we could have a look at that. Don't worry.
Yeah.
That's okay. We've gone over them. We'll see more. Looks like, yeah, we're coming up on some rocks. Not sampleable.
Yeah, they're just a little too big.
Yep. We could get some nice picture if we could, just on the surface of who's there. Nice squat lobster. Zoanthids? What do you think?
That frog?
The
The white thing?
yeah.
Yeah.
Zoanthids?
Yeah, like the one that we found yesterday in the big rock.
Did you get? We didn't sample them.
No.
I don't know how we could.
They shouldn't be very attached so strong.
Would you like some? Okay. We'll just get a we'll just get a nice image.
Yeah.
Yeah. Okay, that's great. Okay, and the rock over to the right. We got one on a clam last night. We don't know. We're interested if these sea anemones might have bacteria associated with their tentacles. Our collaborator at Occidental College in LA, Pasadena, Shana Gafridi, led a paper showing that symbiosis on another kind of anemone. So we're going to see if this one might do the same. Nice shot of the caprella there. So these are a kind of an amphipod. They hold on with their back legs like that, hang out in the current, and you can see their enlarged legs with hooks that are used for grabbing onto their prey. Very common in shallow water, but in fact, great to see them here at 2,700 meters. Okay. I'm just checking if Lisa wants any rocks today. So we can keep looking around and see if there's any rocks we might be able to pick up. I think this will be another field of those feather worms, and we can try and get some nice images of that. We're also a bit suspicious that these worms might also have symbiotic bacteria on their feathery crowns. Maybe if we could sit down and we'll try and get a nice close-up of them when they come out. They live in muddy tubes. So we're just seeing the tops of the tubes there, and then the feathery part is their heads. We'll see how shy they are.
Is anywhere near?
Yeah, that's good. Yeah, there's one starting to come back out now. Yeah, and that one.
And the ship's pulling it toward it, so we're going to get a little bit of
Got it.
Come up and spin around and sit
Yeah,
down.
sounds good.
Alright, come up and we'll gently angle maybe. Alright.
Oh, maybe we should keep going. We might see another patch. Yeah. Sorry, can we just see what all these tubes are here? Or any of this, yeah, what is going on?
Oh, yeah, Xenophyophore.
Lots of the feather worms, and there's, what are those tubes? Oh, this is frenulates. Okay, this is cool. So we'll want to sample this. So frenulates are smaller relatives of the big tube worms. slope. We've got a big mound of them up there as well. It looks like they're just restricted to this area. And what's handy is they form these big clusters of intertwined, curled tubes. So we hopefully can get a good grab of them. These are the first frenulates in any number we've seen on the cruise, so that's it's great to see. We expected to see them back at Concepcion, but missed out. Nicely landed there. So these frenulates were the first of this now well-known group discovered about 100 years ago. They also don't have a gut, and they use symbiotic bacteria. They're usually much smaller than the subgroup of Ceboglinids. The overall group's called Ceboglinidae. Within that, there is Vestimentifera, the giant tube worms we commonly see at Seeps and Bents. But then sediments all over the world have frenulates, wherever there's some hydrogen sulfide. And they are commonly seen at methane seeps, but often overlooked because they're not as visually spectacular as the Vestimentifera. Yeah, we'll have a bit of a nice look and zoom over the tubes and see if there's anybody home. They've got a distinctive wiggle and coil in the tubes. And that's definitely them. There's a nice featherworm coming out, so we've got a mixture of featherworm, sandy tubes, muddy tubes, and coily frenulate tubes there. They're pretty small, and I can tell you they're a dissection challenge getting them out. Prelate skeleton shrimp hanging out on them as well. Oh, we got some snails in there, that's good. So that's Provana. Lots of Provana. That's a nice view of a Provana there on the top of the tube. Often the frenulates don't really come out of their tubes. They do have tentacles, but usually they're retracted. We might get lucky. Could we get an image with the lasers on, just to get a sense of scale?
Sure.
Thanks. There we go. So you can see how small these tubes are. That's 10 centimeters between the beams compared to the big tube worms we've seen the rest of this cruise. Okay. I think a couple of grabs should work. Give us plenty to play with. Into a bio box or a tube. Yeah, they'll be all matted and go down into the sediment. We'll see how they come out. Thanks, Doug. Oh, yeah. So they go down deep into the sediment because they're after the sulfide. Does look like ramen. Yeah. Nice. Look at that. That's great. I think biobox.
Yeah. Okay. Would you want a close-up of this? Are you happy with
Yeah,
this?
close-up would be nice. Thank you.
Oh, wow.
Yeah, they're inside. You can see the bodies. The red. Like their vestimentiferan relatives, they have hemoglobin. And the trick to getting them out is you kind of hold them with a forceps and with another flat forceps slide and you push them out. The tubes are very hard to cut open, so we kind of squeeze them out. All right. Thank you. Those tubes are long. They could be a meter.
Forward, Doug.
Nope, that's good. Oh, my thread is caught.
Nice Urchin,
Another grab would be good too, please, Doug. Yeah. Whoa, look how long those tubes are. Lisa's suggesting these could be small Vestimentifera, and she could well be right there. There is a group like this in the West Pacific that have little wiggly tubes called Alasia. Yeah, these are big. Big but long and thin. Okay, thank you. I don't have one.
You want another sheet?
I will delete this stuff.
Oh. They're sticking.
It's still stuck.
It looks like it's just free now. They're resting under the bumper a little bit.
You can cross them into the other side. We're not short of space.
Yeah, start closing the buttons.
One shot.
Okay. See how it's kind of stuck on there?
Yeah.
That's what I was trying to do.
Gotcha.
Fantastic.
Alright, let's do it.
I just saw a creature. Might see if it's still there.
Which one is that? Yes. Oh, that's good. That's good.
Alright. What'd you see, Greg?
It was a little pink Xenophyophore over behind this bush. I don't see it now.
It's not that. Oh, that's now.
It was on the ground, but it was further back.
Further back.
So. Okay, it's not here. I'm going to hand over to Eulogio, our cruise chief scientist, and talk to you later. Bye. Okay, thanks.
Brief
There you go.
goodbye.
Thanks, Greg.
All right. I think the goal is some rocks. Rocks for Lisa. Rocks for Lisa. Push goes now.
Are we still supposed to be on deck for two?
Yeah.
Okay. Yeah. Right around here. Right around here.
One out.
I just learned about your Dive Island videos. I just learned about your Dive Island videos. It's like, people have like highlight reels of every dive.
I've done them before, but then just for fun and gotcha. But that's roughly, I just gave her the footage.
These three are Marla.
But she very much a lot of footage. Yeah, but that's why I do it at the same time as the Divestream stuff.
Muy bien, buenos días. Agradecemos a Greg, nuestro investigador del grupo que nos acompaña en esta expedición de la Universidad de California, experto en biodiversidad marina. Y en esta última hora, porque a las 12 ya comenzamos la subida del robot, terminaríamos con este dive, el penúltimo del día de hoy, el penúltimo de toda la expedición. Nos encontramos en el sector de Isla Mocha, a 2.747 metros de profundidad. Hemos estado mapeando, levantando información del fondo marino de estos ecosistemas quimiosintéticos sumamente diversos, altamente interesantes, con una biodiversidad totalmente desconocida, nueva, muy llamativa, donde es posible ver adaptaciones de organismos a las condiciones que presentan este tipo de ecosistemas. Una de las características más relevantes, la presencia de metano en el agua a través de burbujas, pero también en los sedimentos, lo que genera una adaptación distinta de los organismos para poder soportar y tolerar condiciones distintas a las que generalmente uno suele ver.
Rocks?
There's a rock.
Is that what we're looking for, right?
What size, Dara?
That's what we want, right?
Yeah.
I'm just double checking. That's what we want. So I don't know if these.
Yeah.
Is that a rock or a dirt? That's a rock?
We can check. I think we can check. Looks like rock, but sometimes when you put the finger, it's not rock. Can we check? I think so. Looks like.
Test this one a little bit.
With the crab. Bueno, vamos a recolectar una roca, que es uno de los objetivos de uno de los grupos de investigadores que se encuentra a bordo. Las rocas actúan como sustrato biológico para muchos invertebrados que encuentran en ella un lugar de refugio, de asentamiento, de hábitat. Pequeños organismos milimétricos o de solo un par de centímetros utilizan las rocas para vivir, para protegerse. El análisis posterior permite descubrir una gran cantidad de organismos con diferentes tipos de adaptaciones. Por lo tanto, estamos recolectando rocas para conocer la biodiversidad asociada a estas rocas. Se recolectan entre 4 y 5 rocas por inmersión del rock. También se hacen algunos otros estudios asociados con el contenido de ácidos grasos de los organismos que habitan ahí, estudios genéticos también, y del mismo modo análisis de isótopos estables. En este caso vemos una roca con presencia de, al parecer, tubos de poliquetos sobre ella. Y bueno, vamos a tratar de recolectarla de acuerdo al tamaño, si lo permite. Si se dan cuenta, bueno, la roca posee también mucho sedimento, mucha arena. Y bueno, el sedimento también es un hábitat muy importante para un grupo importante de animales. Es en donde encontramos la macrofauna bentónica y la meiofauna bentónica, organismos que presentan tamaños en el orden de menores a 1 milímetro en el caso de la meiofauna, y entre un par de milímetros y 1 a 2 centímetros en el caso de la macrofauna. Justamente en esta expedición también se está realizando una investigación a través de la toma de muestras de sedimento para abordar la composición, la abundancia y la diversidad de estos grupos infaunales, porque viven enterrados en el sedimento. Es justamente la línea de investigación que desarrolla el grupo de la Universidad del Paraíso que se encuentra a bordo. asociado también a la recolección de sedimentos para estudiar las comunidades bacterianas, los quistes de diatomeas y también de la caracterización física y química. Las comunidades que viven en el sedimento siempre se asocian a variables ambientales, como el tamaño del grano, la concentración de clorofila, contenido de materia orgánica. BB1.
More, yeah, maybe what do you want? More rock?
Thanks.
Or is that enough?
Which bio boxes?
No,
That was 1A.
1A?
Yes.
Okay.
And más?
Yeah,
Yeah.
we can
Mas?
try another one. Yeah.
Yeah, but I just confirmed with Lisa. It's 3.
She won 3?
Yes, I just confirmed with her.
Just
1B.
Do you want in the same?
It's the same rock. You want it in a different one or the same one?
1B.
1B?
1B.
Okay.
Okay, it's enough. It's enough. It's okay.
It's enough?
Good.
But in the same bio box, 1B, not 1.
1B. Watch out. Last one coming up, man.
Okay, keep going.
Good.
Bueno, como les contábamos, esta recolección de rocas tiene un objetivo de estudiar principalmente la biodiversidad de organismos invertebrados asociadas a este tipo de sustratos. Rocas de distinta naturaleza, obviamente presentan diferentes tipos de organismos. Y del mismo modo también comentábamos que muchas de ellas presentan sedimentos y estos sedimentos también ayudan a crear una heterogeneidad de hábitat, favoreciendo la diversidad.
All right, where to?
Where?
I'll just ask you guys.
Where are we going?
Yeah.
We are here, yeah? We can follow in that direction if we leave. Where we are.
Yeah.
Okay, yeah. They have around that direction. We can walk south or around here.
Okay.
Yeah.
Yeah, she's gonna spin that so we can see what direction.
Oh, it's facing north.
That's north?
Ah, okay, so that way. Yeah,
Back north? Okay.
that way.
So you wanna go back up? Yeah.
This is where we are.
Following this line.
Okay. So straight north.
Is that east to west line an artifact with a mapping, or is that, which one? The kind of one that goes all the way across the street. Oh, this? Those are definitely the different mapping patches. This? Yeah. Yeah, that's definitely mapping artifacts. What do you have? That's interesting.
Roger RV, got an updated waypoint. Copy that.
Bueno, ahí vamos a mencionar un tema que creo que durante la expedición no ha sido comentada, que tiene que ver con las autorizaciones para realizar estas expediciones. Gracias a el Decreto Supremo número 711, que regula las actividades de investigación en aguas de jurisdicción nacional, hemos podido realizar esta investigación, ya que el Servicio Hidrográfico y Oceanográfico de la Armada es quien autoriza todo tipo de investigación científica realizada no solamente por instituciones chilenas, sino que también por instituciones internacionales, a través de la gestión que se realiza incluso a través de canales diplomáticos. Es justamente este servicio de la Armada quien primero acoge la solicitud, posteriormente revisa los antecedentes y la naturaleza de la investigación, poniendo énfasis en la protección del ambiente marino, la no áreas marinas protegidas, territorios de interés comercial, áreas de manejo de recursos bentónicos y también de comunidades locales y comunidades indígenas. Una vez que son revisados los antecedentes técnicos, en el sentido de evaluar, por ejemplo, los métodos de muestreo, los instrumentos, las vías de navegación y los lugares, se autoriza finalmente la expedición y se evacúa una resolución que nos autoriza para poder realizar la toma de muestras, ya sea sedimentos, rocas, agua de mar y muestras biológicas. Eso por una parte, ya que la autorización para poder obtener principalmente las muestras biológicas, es decir, los animales, es resorte del servicio de la Subsecretaría de Pesca a través del documento Pesca de Investigación, que no es una pesca comercial ni extractiva, sino que lleva el nombre de Pesca de Investigación, pero tiene objetivos científicos. Y en este sentido se privilegia la calidad en lugar de la cantidad, es decir, no extraer volúmenes comerciales de estos animales, sino que muestras justamente para su estudio e identificación. Es así como estas investigaciones permiten a investigadores internacionales, en el caso de que solo hubiera investigadores internacionales, a realizar estos estudios y tomar las muestras para los objetivos que se señalan en los proyectos. Por lo tanto, también a través de mi persona como jefe científico de la expedición agradecemos las gestiones del Servicio Hidrográfico y Oceanográfico de la Armada y de la Subsecretaría de Pesca por otorgarnos estos permisos que sin duda contribuyen al conocimiento de nuestro mar, al conocimiento físico, biológico, químico de nuestra columna de agua, de nuestro fondo marino. Sin estos permisos no podríamos realizar estas investigaciones. Por lo tanto, son de vital importancia y es bueno también reconocerlos. Se podría comentar que sí, pueden ser un poquito engorrosos algunas veces, lentos en su tramitación, pero bueno, es parte ya de nuestra idiosincrasia nacional y ya estamos un poco acostumbrados a eso. De hecho, como parte del decreto 711, se genera un acuerdo entre la Universidad de Boston, el Schmidt Ocean Institute y la Armada de Chile. Es decir, se genera una resolución que establece y que otorga deberes y derechos a ambas partes, y deben haber cumplimientos en los procedimientos, siguiendo una serie de protocolos en todos los procedimientos que se realizan, desde la recalada, el zarpe y los trabajos técnicos que se realizan ahora. De hecho, se encuentra con nosotros a bordo durante toda la expedición un observador nacional, que es el señor Diego San Martín, quien justamente vela por el cumplimiento en regla de todas las actividades que se estipulan en estos permisos y decretos. Él es el representante de la nación técnicamente en este barco extranjero y revisa que nuestros procedimientos estén acorde con lo que hemos acordado. Close, if possible. There's a worm. Okay. Oh, no, no. No worms.
No worms?
No. It's just the mouth. Looks like sometimes like a worm, but it's not a worm.
It's just a mouth, huh?
Sometimes when it's common, some worms live inside.
Oh, okay.
There's some specific species, a specific relationship between some anemones and some worms.
Okay.
Some specific worm species live just in that kind of anemone.
Gotcha. Yeah, cool.
In that case, well, perhaps the worms are in another side.
No one's
Yeah, perhaps he went to the supermarket or something.
It's on vacation.
Okay, thank you. How you remember in previous expedition from the Oceanic Island, all the worms with the sea urchin, probably the Rapa Nui cruise, Salasimus cruise. You remember the urchin below the sea urchin, always the polychaete.
One in
Only
there.
one,
Yeah.
around there. This probably he live around it.
Similar. Okay, that's cool.
characterize your RV, you got an updated waypoint?
Copy that.
I will make it.
Please.
Yeah, the same. Close up this. Alright. I don't know where's that. Take a picture. Okay, thank you. Thank you.
Uy, yeah.
Ah, yeah, we need to collect that if it's possible. Yeah. Wanted.
No.
Wanted the molecules.
Keep that. You all deal with this.
Still there?
There's extra jars or whatever jars. you
I
gotta
see.
Mocho.
Section 1, no, 8, no.
One?
1 or 2, 8, no.
No, because it's full.
1 or 2.
One?
Yeah, 1.
Okay.
Succiona. Saca.
I don't even see it on the sit. Oh, that's right. Okay. I see it now.
Out.
It's just behind you. You bring this in a little bit so I can get some, I can see it coming a little bit better. Everything's fighting us, dog.
Where is that?
I hear. It's here. This one.
Okay, you know.
There
You know.
it is. Watch it.
Bueno, comentaremos que estamos tratando de recolectar un poliqueto pelágico a solicitud del Dr. Greg Rose, experto mundial de este grupo animal. Es complejo pillarlos, nadan, se mueven. También el movimiento del agua, las corrientes lo hacen moverse y bueno, resulta complejo. El grupo de poliquetos pelagicos no es muy diverso, no hay muchas familias de poliquetos pelágicos. Es mucho más diverso y abundante el grupo de poliquetos bentónicos que viven en el fondo, entre las rocas, en los sedimentos. Tampoco hay muchos especialistas de poliquetos pelágicos en el mundo. Muchos de ellos son parte del plancton, del zooplancton, poliquetos holoplanctónicos. Por ejemplo, ahí tenemos la familia Tomopteridae, Lopadorrhynchidae, Iospilidae, Alciopidae. Y bueno, aquí tenemos un poliqueto que nos interesa mucho por encontrarse en este hábitat de filtración de metano bastante profundo. Un poliqueto que no sería que al parecer no formaría parte del zooplancton por encontrarse más asociado a esta comunidad. Al parecer lo logramos capturar con la bomba de succión. Así que va a estar feliz el Dr. Rose y para posteriormente poder estudiarlo.
It's in jar 1. I just switched to jar 2. It's all inside out. I think it is.
We got it?
We did it.
Still on deck for 2 p.m., right? Yeah. You guys can talk about it.
Les contamos que nos queda solamente el día de hoy. Hoy día terminamos los dive, los muestreos, como parte de este crucero que lleva por título Cañones submarinos, filtraciones de metano y chimeneas hidrotermales en el margen de Chile continental. Estaremos llegando el día 4 a Puerto Montt y este primer crucero, esta primera etapa culmina para después seguir en una segunda etapa con objetivos similares, con un grupo renovado de científicos, donde destacó al grupo de investigadores chilenos que realizarán los estudios bentónicos de sedimentos, de macrofauna bentónica, de meiofauna bentónica, de química, comunidades bacterianas, pero también que van a cargo de la recolección de la fauna. Nos interesa mucho obtener colecciones de invertebrados y peces en la zona de los cañones patagónicos y sobre todo si logramos encontrar esta chimenea hidrotermal activa en la zona de la triple unión de placas tectónicas que se encuentra en el sector de la península de Taitao a más o menos unos 3.000 metros de profundidad. Para ello, la Universidad del Paraíso está colaborando con el Museo de Historia Natural de Santiago a través de la participación del doctor Jorge Pérez Schulteis, curador del museo a cargo de las colecciones de crustáceos y otros invertebrados. También la Universidad del Paraíso colabora con el doctor Matthew Lee, que también se embarcará en esta etapa, experto en lo que es el estudio del meioventos. Y el grupo también lo cierra de investigadores que colaboran con la Universidad del Paraíso. También participa en esta segunda etapa el doctor Américo Montiel de la Universidad de Magallanes, quien desarrollará toda la investigación de la biología de los sedimentos, estudiando la macrofauna y tomando muestras para el estudio químico. También apoyándolos está la señorita Josefa Araya y Tania Figueroa, ambas apoyando este grupo de investigadores chilenos. Este crucero comienza el 6 de noviembre y termina en diciembre, la primera semana, en Punta Arenas. Make it close up, please.
It's a sea cucumber.
Okay, can make it. Okay, thank you. Can this one?
Yeah, most of the time.
Say something.
Yeah.
Don't worry, it's fine. Yeah, put it out there.
Yeah, it's gotta be.
Yeah.
further out.
Out there in the middle. Can we collect that one, please?
What did I?
It's an isopod.
It's a suction sample?
Yes, maybe 6. Should be fine. 6. Bueno, vamos a recolectar ahora un isópodo. Estos organismos dorsoventralmente aplanados son crustáceos del orden Peracárida, pariente de los anfípodos. Comúnmente también los anfípodos se suelen conocer como las pulgas de mar, pero este es un isópodo. Los isópodos, por ejemplo, para que el mundo los relacione, son también los chanchitos de tierra que tenemos en los jardines, en la tierra. Bueno, este es un isópodo con antenas largas, patas largas, distinto, de profundidad, de color blanco, acostumbrados a vivir en ausencia de luz. Adquieren mayor tamaño, con mayor profundidad, y va a ser muy importante poder recolectar este organismo. No es común encontrarlos, no se tienen muchos ejemplares de este tipo de animales en los museos y en las universidades chilenas. De hecho, tampoco existen muchos expertos que estudien estos grupos. De ahí también la importancia de motivar a quienes nos están viendo, principalmente los jóvenes, los estudiantes, para justamente desarrollar la taxonomía de estos grupos desconocidos. Thank you.
There he is.
Perfect, thank you. Only one, if we can find one more rock, only one more.
The rock, eh?
If we can find.
We don't have time to make it over to those like southeastern points.
Bueno, quienes nos han seguido durante todos estos días, yo creo que se han dado cuenta de la alta biodiversidad marina que presentan nuestros ecosistemas profundos. Hay una gran variedad de organismos, pepinos, nidarios, corales, estrellas, en fin. Sin embargo, existe un alto desconocimiento taxonómico, biológico, fisiológico de todos estos organismos. Esperamos haber entusiasmado y motivado a los jóvenes, principalmente a los estudiantes, a conocer más sobre la vida marina, sobre la biología, a aprender más. No es fácil, no es común tampoco que mucha gente siga más adelante estudios que desarrollen las líneas de la identificación, de la taxonomía. Hoy en día es necesario conocer sobre el patrimonio natural que tiene nuestro país y no solamente interesarse por los animales grandes como mamíferos, aves o las plantas, sino que también por estos pequeños organismos que ustedes mismos se han dado cuenta son de una gran belleza y de formas muy interesantes. también uno de los objetivos relevantes de todas estas investigaciones es generar colecciones que posteriormente queden depositadas en los museos, como una forma de educar, de difundir, de dar a conocer nuestro patrimonio natural. Muchos de estos organismos también son posteriormente estudiados y analizados en los laboratorios de acá científicos, pero es importante que el acceso de estas colecciones sea abierto, y para ello están las universidades y principalmente los museos. Esperamos durante estos días, con todo lo que hemos mostrado, haber podido motivar. Hemos hecho también transmisiones en directo a través de los ship to shore a los colegios en zonas incluso rurales. Just a close up. Porque claro, bueno, es sumamente, sin estos mecanismos, sin estas transmisiones, es casi imposible ver el fondo marino. Hoy en día existen los documentales, pero creo que estamos marcando un hito importante, ya que estamos mostrando la vida marina que hay en el fondo marino de nuestro país, de Chile. Generalmente uno está acostumbrado a ver estos documentales en la televisión que muestran el fondo marino de otros países, pero en esta oportunidad hemos mostrado nuestra fauna, nuestra biodiversidad. Y creo que ha sido muy interesante. Acá, por ejemplo, ahora vemos un organismo, un hemicordado. Estos organismos, por ejemplo, son muy evolucionados, están más emparentados con las ascidias. También se le llaman gusanos biota, pertenecen al grupo Enteromneusta. Y fíjense la forma, los colores, las adaptaciones, son organismos muy extraños. Me atrevería a decir que estos nunca habían sido documentados, nunca habían sido obtenidas imágenes de estos organismos en Chile. Sí se han obtenido imágenes en otros océanos, pero en aguas nacionales frente a Chile es primera vez, y a través de esta expedición se han podido mostrar este tipo de organismos. También los xenoturbelos, que parecen como globos desinflados y que tienen una posición taxonómica actualmente discutida. No se sabe también a qué grupo pertenecen. También lo hemos podido mostrar. Durante la mañana también han aparecido estos gastrópodos sin concha, parecidos a erizos, que también han sido muy interesantes. Realmente ha sido una clase de zoología marina de 3 semanas, fascinante, completísima, más encima con la presencia de expertos que nos han enseñado y nos han mostrado y nos han explicado cómo son, qué son, cómo habitan, qué adaptaciones tienen. De hecho, ha sido muy divertido darnos cuenta de cómo algunos colegas acá que no son biólogos, que son geólogos o físicos, es que es imposible no quedar ajeno a esta maravilla, a esta belleza que muestran estos organismos, los colores, las formas. Entonces muchos de ellos se han interesado y les ha gustado mucho tener la oportunidad de ver en directo estos organismos. What size is the starfish? Do you have the lasers?
I'll give you the lasers here right now. There you go.
Gauge.
Coral quivers works for that, or it's too big? What do you think?
For what?
Would the starfish coral quiver work for that?
Maybe.
Yeah, maybe.
If not, we have 4 bio boxes open
We
in
need-
9 minutes, so I don't think we're gonna fill all the bio boxes. We can put in the bio boxes if we want as well.
Okay.
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 Correct. Yeah, last second.
spent 7 years waiting for the dust to settle, Doug.
Agradecemos también los comentarios que mediante el chat en vivo la gente nos ha hecho llegar. Efectivamente, bueno, el Smith Ocean Institute también tiene como objetivo principal difundir todo lo que estamos haciendo y el mecanismo es justamente las transmisiones en directo a través de las plataformas de internet, del canal, de la página de YouTube. Por lo tanto, son canales abiertos y mientras más gente difunda estos videos, más gente conoce la naturaleza del mar chileno. Así que también los llamamos a difundir, a traspasar estos links. Y bueno, agradecemos los comentarios que nos han hecho. Justamente el conocimiento de la biodiversidad es el mecanismo, es el argumento más fuerte para poder proteger posteriormente si estos ecosistemas reúnen los criterios para finalmente gestionar posibles medidas de conservación o generación de parques marinos. Siempre es bueno, mejor proteger lo que se conoce y no proteger lo que no se conoce. Al menos eso debiera ser la premisa, la prioridad. Okay, if we can, thank you.
Rock.
We have time.
We have
The rest of the time.
4 minutes.
4 minutes.
Okay, now sorry. Can I ask, kindly ask the skin bottom to be closed?
This can, yes.
Yeah. Oh, retracting this skin as fast as we can go, which will take you back to 4 minutes.
Keep the red light.
Party.
Flag line.
4?
We were doing 2 of them. 2 Niskins or just 1?
No, this one.
Just 1?
Thank you.
Okay.
Which one?
Bien, bueno, nos despedimos ya que debemos subir a la superficie, pero nos vemos más tarde. Nos queda un último dive, así que atentos a la transmisión. Vamos a realizar un último dive de la expedición, eso de la tarde, así que ahí también vamos a estar mostrando más fondo marino de Chile, más biodiversidad, más animales. Nos despedimos, gracias.
Where is it gonna take weight one?
It's really nice. It's really good here. Just play with it. Some others. We don't show. Looks like a beautiful monster.
Beautiful monster.
Beautiful monster.
I know cool monster.
Oh, monster.
Can you see the
Can't blast it.
Can you make it less close up?
Is it? Urchin. If you wanna do your sign off right there, we're gonna start coming up.
I know, yeah, we already.
Do you wanna do it now?
Thank you.
You ready? Yeah. Alright. Brad Jarvie,
Yeah, go ahead.
that's gonna be the ROV lead at the bottom.
Okay.
Okay, ROV is coming off for copy.
Tight for a second.
It's all right. No. 18 days. I suppose.
Oh, good.
Okay.
Juliana got a good screen grab on the,
He's coming out, yeah.
sea log.
Thank you.
You ready, Ben? Yes. Auto done. Don't know where I went, which picture. TR will be off bottom bridge.
Go ahead, Doug.
That's us on the recovery.
Okay, cheers, man.
Got exit if you wanna.
Thanks, Dave.
Okay, that's the ROV off bottom. We will continue streaming our ascent, and then we'll have a short period on deck to do some gravity cores and moving to another location. And then we will have our final dive of this leg later this evening.